Keep spawning culture. Hey y'all, it's Andrew Reed and Samantha Reed with Mossy Creek Mushrooms, the MushRune Cast. Today we've got some cool stuff to talk about. I'm pretty excited, which I'll do the introduction here in just a moment. But we are going to have Josh McGinnis with EverymanBio on. So
I'm excited.
Me too. He does
Hopefully
cool
Josh
stuff.
is as excited too.
I would definitely hope so, right? We're going to give it a little bit of time for the audience to build up before we start getting into the conversation. What's up, Michael? Glad to have you on with us. Again, anyone who is on Instagram, I would like to remind you that if you want to participate in questions with Josh and Samantha and myself, please come over to YouTube or X, because I can see it and respond to you automatically. But if you're on Instagram, I have to go back and check it manually, which means I have to leave the digital studio to do so. So it only happens occasionally. So I'd like to remind anyone on Instagram to head over to X or YouTube if you want to participate. Either way, we're glad you're with us. I've got a couple people on X and some more joining. If you guys on X will please give me a quick, just one comment or so, just let me know that the X stream is working. I'd really appreciate it. So excited for this episode, restaurant feedback. We could probably do that a little bit later. I think Josh is going to leave us sometime about 6 or so. Samantha and I usually go around 6:37. So I think we'll be able to get into some Q&A and stuff, that kind of stuff. Yo, it's BTV Farms. Awesome, Michael. Thank you for letting us know. Appreciate that. That was from X. Afternoon, Power Man. So we got some cool things, like I said, to talk about. Samantha, is there anything that you're going to want to cover today? I didn't ask her yet.
Just here to go with the flow.
You squid-faced. The squid face. That's the Morty face for Rick and Morty, if I ever saw one. Hey, Ed Grand's with us. Hello from Chile, Thailand. Had dew on your motorcycle. Chile, Thailand, dew. We're frozen. I spilled a little bit of water outside, and it instantly froze. Like, it's so cold here. Even in this room. I have the heater on. Samantha and I are bundled up. We have incubation going. We have heaters in there. And the HRV going. And it is still chilly, even with all the studio lights and stuff going.
Heaters at our feet. And
yep.
I have to be careful if I'm going through if I walk into the grow room and it's slightly, the water on the floor is slightly sticky. It's not like puddled or anything, but if I walk through there and then go outside, like my feet will stick onto the back dock. Yep.
Yeah, I was experiencing the same thing. I was on that. We have a metal plate that traverses a line in the concrete, the seam, and especially on that metal plate, I was stepping. I was having to my feet were almost instantly freezing to it. So
it's kind of funny.
Oh yeah,
we're not used to this kind of cold, to be honest.
No, we are not. It is. I will say, I've had one tiny little shiitake that was starting to grow off a block. That I was like, oh man, I'm gonna get spore print off this thing, and it has stayed this size for days now.
It's stalled.
It is done. It is given up. The ghost. Aww. It is not working. That's just not gonna do nothing no more. The only thing I've got going crazy right now, just check it
It's
out.
Enoki.
My white enoki is going insane. So we're definitely thankfully I was able to send a message to the distributors and be like, hey, you guys want a white enoki next week? Because I am going to have plenty of that.
You know, I'd really like to go down a little bit on the substrate and give it a bit more room. See what.
I think that would actually work. This was made on day 335. That may be reversed on the. I'm not sure. But anyways, it's this was day 335, which is what it'll be 60 days and 3 days because last year was 366 days.
It's a leap year.
Yep. So, we've got. Golly, I'm glad I did that white enoki. We just did it as a test, but we've got quite a few bags of it going.
There's like 3 racks back behind us. Oh, we should have moved those.
I did right here, but you can't see them as well because I've put them in front of the sign. Yeah, but there's like white enoki all in these bags behind us, and it's growing like crazy.
Good job. That's a nice forethought.
I thought about that. Yeah, that's one of the janky shelves. We got a couple of shelves that are starting to lose the rubber on the casters. So.
Yeah, need some maintenance.
Frank says, hello, my favorite mush couple. Well, hello, Frank.
Hi.
I said last week that I didn't think it would snow in Florida. Ended up with 9 inches and single-digit temps.
Wow.
I will say my buddy Foxy that runs the EverymanBio subreddit told me it was snowing where he lived, and he's like, what is happening?
We're as surprised as you are, but I'm sure you're even more surprised in Florida.
Lost power and had to bust the hurricane generator out at 4 a.m. to keep the pipes from freezing. We have got pipes freezing right now. Our washer is down. Our warehouse sink is down. Now the hookup to the production Thor, both our Thors is down.
It's frozen.
Completely frozen solid. We have an icicle because apparently we had a drip. We have an icicle that's about 3 feet long behind the sink in the warehouse.
Well, and the faucet, the part
Samantha drew
that is the faucet.
I told her it was frozen, and she's like, I'm just gonna go test it. I don't believe it. She pulled the lever, and the faucet popped out.
It's not that I didn't believe you. I went to go put water in the hot kettle to make morning tea, whatever, and then it just came off. So we'll see what happens with that.
That does happen. Hey guys, again, I see you people server. Hey guys, love your culture packs. You're not selling them together anymore. We absolutely are, and we do absolutely do international shipping to most countries with biosanitary certificates. So you have to have that kind of stuff. But anyone who's on Instagram, please join us on YouTube or X, because that is where you will actually be able to chat with us back and forth. So. In
fact, I learned some really cool new harmony codes today for that exact reason.
Oh, did you?
Yep.
Oh, neat. So I got a shiitake block in my room right now with a single mushroom stunted just like yours. Maybe they were rebelling against us. It's the cold, man. They rebel against the cold. They just can't handle it. Let's see. Sorry, I've got a couple of things I'm trying to pull up here. I'm trying to prepare a couple of share screens for you guys. One of the cool things about what Josh does or has on his YouTube channel is some time lapses, and I'm trying to get some of those where we can show those while we talk with him, because I think they're really cool. I've tried my hand at time lapses and I've burned through 2 or 3 cameras doing so, but I've not really been successful at the mold ones. So I'm hopefully gonna get a chance to pick his brain about how do you take a time lapse of a petri dish without it like fogging up really bad. So
that's a really good trick of the trade.
Yeah, mine always fog up within like seconds. So
it's because you're standing there staring at them like this.
I think so.
Hey, come on, grow.
I think it's more the light for sure, but well, it's about 10 minutes in. I think we can just go ahead and start. Josh, you good? Can you give me a thumbs up or a thumbs down? Thumbs up. Okay, cool. All right, guys. So Josh McGinnis, you guys may remember, came down and did a like a little bio prospecting trip with me. We hit a cave. We hit the old grain. I don't know what you would call it in the basement, like a grain dump. storage
Silo?
base. It's not really a silo because the silo is above ground. But anyways, we hit that in a cave in a lime yard, like a lime pool and stuff. He bioprospects molds, and he's always excited about the weirder and the more rare, the better. And I think he does some bacteria and stuff as well. But I mean, honestly, we'll let him introduce himself. Without further ado, it's Josh McGinnis. What's up, Josh?
Hey guys, Andrew, Sam.
How you
Good to
doing?
see you guys? Not too bad, not too bad. It's kind of surreal being here because just like a quick story, when I first was getting started, like a couple of years before COVID, which is hard to believe, it's already been like 2 years since.
I know, right?
I was like searching YouTube for different techniques on growing mushrooms because when I first started doing this stuff, I had zero experience, which I could talk about, going from zero to publishing papers and things, doing things like that. I was like, I came across your channel and I was just sort of enamored by one, the huge following you had. In fact, I gotta admit, it was kind of hard to even watch it at first because I was like, oh, this guy's so far ahead of me. I'm never gonna be at that level.
Gosh.
So it's a little surreal now to be like hanging out with you, doing bioprospecting trips, and getting invited to your live stream. So it's cool.
I appreciate that. Though I will say, I constantly look at other people's channels, and I feel the exact same way. When Mike Crow blew up and passed right by me, I was like, my gosh, I'll never be as good as him. So
I still feel that way.
Do what?
I still feel that way about different people, and then it's weird when you start getting people come up to you and say the same thing to you, and you're like, I don't know anything. So
that's where I stand too. I'm like, man, I barely know anything. I know just enough to know how little I know, but at the same time, yeah, like it was strange. We went to the Oklahoma Mushroom Festival. Unfortunately, we couldn't go this last year, but the year before, and when I was 2 steps out of the car. And somebody was like, Mossy Creek mushrooms? And I was like, what? They're like, Andrew. And I was like, and then it was like another 2 steps. Someone else recognized me. And I was like, whoa, am I a celebrity? I feel kind of like a celebrity.
you have a very recognizable face. It's the beard and then your branding, very unique branding.
There is that.
Well,
we showed up too with new T-shirts. So Samantha and I, and then even Ben Dooling had made T-shirts specifically for the occasion. So we had T-shirts with like some of the bioprospector T-shirt and stuff that we have the images on the back, and then Moss Creek logos on the front. But nobody could see that because we were wearing sweatshirts and stuff because it was so cold when we were in Oklahoma. But
it's true.
So he said, Josh, live from the old growth forest.
Yeah. I've got my Starlink set up over here, and
got your Starlink camp. I like it. I could deal with it. I actually thought about that. Like, how cool would it be to be able to go up into the old growth? But man, the power requirements would be unreal.
Yeah.
But yeah. Oh my gosh, my brain just completely blanked. I'm sorry. My ADD kicked in. That's why I asked for coffee. I told Samantha that I was like, I'm going to need coffee today because I don't know what's going on, but I slept like crazy and I still cannot wake up today. So
funny enough, it slows your brain down.
What?
It's the cold. I think it's the cold.
It's got to be the cold. It's got to be the cold. Oh, I was going to ask you to just do a short introduction of yourself, if you will. I tried my best to give you a good one, but I always like to give somebody an opportunity to fix what I've started.
Greatness speaks for itself.
Well, I like to tell people that like I don't come from a biology background whatsoever. I spent most of my career, if you want to call it that, in software engineering. And that's kind of what I've always done for like the last 15 years. And that's what I do to pay the bills. But I would say like 6 or 7 years ago, I sort of had like this midlife crisis where I got burnt out on what I was doing with my day-to-day. I was like, all I've done is like write code for the last, decade plus. And I just couldn't do it any longer. And I didn't know what I wanted to do. But I knew that I always had a love for biology. And I wanted to do something that would connect me to something living. Working behind a computer all the time, it's like, so you're working with something inanimate, and I think people can connect with this idea of like being closer to nature being closer to stuff that's living, whether it's farming or it's growing mushrooms or something like that.
Yeah.
So I just started growing mushrooms again. I'd grown them once before, and then I grew some shiitakes, which are really hard. I don't recommend you start with shiitakes as your first mushroom to grow.
Yeah.
Are you guys familiar with that? You have some experience with?
Oh yeah. It's in fact, that's my big. A lot of people that come into mushroom farming, unsurprisingly, want to grow shiitake because it's so well known. I mean, it's one of the few mushrooms people know other than portobellos. But it's I always tell people that it's not really if as a beginner grower, stay away from it because it's a little bit more hard, a little bit more difficult to grow. But beyond that, it's also just not economical unless you're big. So,
I grew them using the PF Tech. You familiar with PF Tech?
Oh yeah,
Actually, that actually worked for shiitakes.
Wait, did you do it with liquid culture or spores?
Yes.
Okay.
Yeah, liquid cultures. Yeah. They were very sad looking shiitakes. I mean, they came out very puny and like one just shot out the side and that was it.
I grew oysters my first time around and I was so proud of them. And now I go back and I found the picture the other day and they were the leggiest, most stretched out looking oysters I'd ever seen. So I get it for sure.
Yeah. But I started growing mushrooms and I started growing everything I could possibly get my hands on. And then I realized pretty early on, because I'm kind of a nerd. I'm not kind of a nerd. I really am a huge nerd about pretty much any kind of, I love learning. So any kind of technology, any kind of science I'm super interested in. And I really thought long and hard, do I want to become a grower? And I was like, that's not really the direction I want to go in. I mean, there's so many directions you can go in with this stuff. And I was like, well, I want to do something in science. And I don't really know what to do. And I was also struggling with I lived in at the time, I live in Virginia now, but I lived in South Central LA, which is a very urban part of Los Angeles. And I didn't live close to mushrooms. I was watching all the people I admired on Instagram and all social media, going out and finding mushrooms in the woods like these behind me. And I'm like, I don't live in that kind of environment. If where I lived, if I wanted to drive to the woods, it would take me a 2-hour drive to go to the woods. And it's also very dry because it's Southern California. Now there are mushrooms. I mean, there were mushrooms in front of my yard. I used to have sprinklers and I'd sprinkle my yard and I'd get, all kinds of various things growing there. But I really lacked access to diversity of mushrooms. And then, but I started culturing them anyways, and not knowing where it was going to go. And one day, as I was culturing them, I kept getting contaminant mold growing on my plate. And obviously, if you're a mushroom grower too, you're familiar with mold because most mushroom growers are that's the bane of their existence, right? They're constantly fighting off mold.
Oh yeah.
And then this light bulb kind of went off in my head that instead of trying to avoid molds, why don't I just embrace them and start studying molds? I didn't really see anybody in the community that was really going really deep on molds in particular.
Yeah.
And the more that, go ahead.
No, I just said, yeah, I was just encouraging you.
And the more I dug into molds, the more I realized one, how important they are to society. We use them to produce all sorts of compounds and chemicals and medicines and food products. And I was like, wow, there's this whole world out there. And then I realized the molds that are being used industrially, there's a couple of dozen species being used. And so this light bulb kind of went off that hey, here's a little niche that I can carve out for myself that will keep me inspired and motivated to keep pursuing science in mycology. And so that's really how I started with molds. And then very soon after that, when I started isolating molds, and then I figured out how to sequence them or DNA barcode them. Yeah. Very quickly, I realized how easy it was to find new species. And I was like, whoa, I didn't realize, I thought finding a new species would be really, really hard and it would require me to travel to far off places in the world. But it turns out you could literally open a plate in the air and something randomly could hit it. And it might be a species that no one's ever,
That's cool.
characterized.
I mean, that's absolutely true. Yeah.
And that's really hard in science, to find an area in science that hasn't been fully explored and exploited without you having to get way, way super technical and, kind of super esoteric. So,
oh yeah,
wow.
I don't even remember how many it was. I mean, I guess we could look it up real quick, but like how many estimated fungal species there are, but
It's a lot.
oh yeah. Fungal species are there. So it says something. There's an estimated between 2.2 and 5 million species of fungi, and there's only about 155,000 species that have been identified so far. So that's a lot of area to cover.
Well, and also just to kind of throw another factoid out there, with every breath you take, on average, you inhale about 100,000 mold spores with every breath. Isn't that
literally
crazy?
a living ocean?
Yeah, so we could talk a lot about. There's a lot of myths, fear about mold. Just mold growing in your house. You might see some if you have a leak or something like that. How do you get rid of mold? Things like that. You can't just putting that out there. You cannot get rid of mold. You can lower the amount of mold growing in a particular area, but so I started getting into molds and very quickly started making some headway there and some science, and started connecting with various scientists at different academic institutions who kind of made me aware that, hey, like, you're kind of onto something here, because if you find a strain of mold that produces a new compound, that could be really, really interesting, because most of academia and most of, commercial entities out there are not doing the kind of research that I'm doing. They're working with known strains that produce a known thing, and they're doing research on that. But, the kind of work where you're going out into the environment, you're looking for new strains that might produce like a novel chemical. Well, that's really interesting, and there just aren't a lot of people doing that. And I could talk about, there's a lot of reasons why. It's not easy, and there's not funding for it, and there's, you have to be self-funded. That's why I kind of call myself an independent scientist. There's a whole Wikipedia page on independent science, but there's lots of noted scientists throughout history that are known as independent scientists, and almost all of them were self-funded because the institutions at the time just didn't see value in their work, so they had to pursue it on their own until they made some big discovery, and then people were like, "Wow, well, why didn't we fund that guy sooner? Why didn't we hire that guy sooner?" That really hasn't changed all that much. It's a very high risk, high reward sort of venture. So nowadays, I've got a collection of about 500 molds that I've DNA barcoded, and I lend that collection out to various research institutions around the world that will screen them for various things. Like, you can't screen for everything all the time, and so different institutions might be like, they're looking for a new cancer drug or new cancer molecules, and so they'll screen it for this very particular type of compound and see if any of them will produce it. And other people will screen it for different things, but to me, it's very exciting, this idea that you can go out into nature and isolate not only some potentially new species, but even if it's not a new species, a new strain might produce some new compound or chemical that hasn't been characterized yet. And that could be really, really valuable to society.
Yeah, absolutely. Well, I mean, I was watching some documentary on production methods during World War II. Completely random, just watching this World War II thing, as most dads do, I guess. It's like the barbecue in World War II is the two stereotypes.
I'm a World War II nut. I love Band of Brothers and Saving Private Ryan.
Dude, so I watched this. I watched this stuff like a lot. Like, I was watching this stuff last night even. But this documentary, they were talking about these production methods, and they literally just kind of slipped into penicillin production. And on that, I noticed that they had flasks on these chain-driven conveyor belts kind of things where they were working, and it was like, that's liquid culture. Holy crap! I reckon they're doing liquid culture back in the 40s for penicillin production. It blew my mind. Like you could even see the pellicle sitting on top of the and it looked like they were literally, those chains were moving and they were kind of, and it's like, oh, that's how they're aerating that stuff, right? They're keeping that liquid moving by just jostling it. It was, in fact, I think let's see, I found one because you were talking about it, but like, oh crap, I really hate it. Okay, StreamYard does awesome, but it does a couple of things. Like this right here was the World War II production, and like they had like so many of these are just liquid culture flasks.
Yeah, so
it's crazy.
Can we talk about that for a second? The process of how this
Please.
works?
Yeah.
Yeah. So, we've all probably been to a brewery. If any of you have ever been to a brewery, you probably see those massive steel tanks in the back, the fermentation tanks.
Mm-hmm.
Those are your flasks blown up to 500 liters or 1,000 liters. Inside of that is a liquid culture with an organism. If it's beer, it's going to be a yeast. But when it's penicillium, it's kind of the same concept. And different organisms will, express those metabolites under varying different conditions. So what'll happen in a lab is like, let's say someone screens one of my strains and they grow it. They're going to grow it on a flask, mostly on grain. And then they're going to take that grain and they're going to put it in a solvent. And depending on the type of molecule, depending on its polarity, that will depend on the type of solvent solution that they use, because that's what's going to allow them to isolate the type of compound. Because actually lots of compounds get produced by fungus when it grows on grain or any kind of liquid culture. Yeah. So once they extract it with solvent, and they'll put it through a series of filtration steps to kind of get it down, and then they'll put it into some fancy machinery, mass spec machines that will actually identify those compounds. So that, like, once they know a strain can produce a compound, then let's say they now want to start working on scaling up the production of that. They'll actually now start trying to figure out what are the growth parameters that this organism and this strain in particular likes that will maximize the yield for that particular compound. For example, does it like to be stirred? Does it like to have light or no light? How much oxygen does it like? What kind of pH does it like? And so at a small scale, they'll create lots and lots of flasks. I mean, I've gone into labs where it's like just shelves and shelves full of flasks, all labeled with their different experiment parameters. And they'll try to find these parameters that maximize the production and all the different variables involved. If you're gonna grow something for commercial, you want to grow it as quickly as possible, maximizing yield, utilizing as cheap a feedstock as possible, because that's going to maximize your process at the end of the day. Mm-hmm. So once they sort of like narrowed that down, then they'll keep doing scale up experiments. They go, okay, we did a 1 liter flask. Now let's do like a 10 liter grow, and a 10 liter bioreactor might be, like a big jug looking thing. They'll do several of those. Yeah. And then like once they've kind of figured that out, then they'll go up to these huge bioreactors that are in warehouses that are like what you see in a brewery, that are like 1,000 liters.
Yeah.
But they still have all the same problems that us independent, growers have to deal with. There's lots of steel piping connecting all of these huge bioreactors. And so between grows, they pump steam through it to sterilize it all.
Yeah.
But there is always going to be occasions where even in a 1,000 liter tank, they get contamination, and the whole thing has to kind of get thrown out, and they've got to start all over again.
Yep.
There's a whole discipline around doing large scale biofermentation of these secondary metabolites. But it kind of all starts with, can you find a strain that produces something kind of, interesting?
And that was one of the things with the penicillin, wasn't it? Is that they actually found like a golden variety or like an amber colored variety that like they were asking people to send samples in. She sent this kind of amber colored penicillin in, and it like was way more productive.
I have a slide on Alexander Fleming. Can I share that with you guys?
Sure. There we go.
All right. Let's see here.
So say I'm not familiar how it works from your end, so.
Can you guys see that?
No. Oh, yeah. Yep. Yeah, we can.
All right, cool.
Wow.
So yeah, I can. There's some cool slides in here. So I can't see you guys now because I'm sharing. So just yell at me.
Yeah, that's fine.
Yeah. So Alexander Fleming, he's obviously the guy who discovered penicillin, and this is a picture of him in him at his desk actually. And he was actually considered a messy guy. In fact, what he was actually studying was bacteria. He noticed in World War I that all of the soldiers that were getting wounded and stuff, there's so many of them were dying from these bacterial infections. So he started researching bacteria.
Huh.
And one day, like he went away for, I think for a long weekend or something. And he was working at what is now St. Mary's Hospital in London on the second floor, I believe. And before he left, he opened up the window to his office to make sure it had air, to circulate over the weekend. And he left one of the plates that he had streaked with bacteria sitting on this desk here. And when he came back into the office on Monday, he was looking through his plates and everything, and he noticed that there was a colony of mold that was growing on this plate that he had streaked. But he also noticed that there was an area, what's called an area of inhibition. I'm going to jump ahead a little bit. Kind of looks like this.
Hey, I did that in college lab, yeah.
Yeah, exactly. Except for instead of these little dots are little pieces of paper that have antibiotics on them. But he actually saw, kind of this ring around the dot where the bacteria wasn't growing. So he's like, that's an interesting observation. But it happened completely by accident. And he didn't know what to do with that. Like, he wasn't studying fungus. He didn't know anything about. He didn't know it was penicillium. He didn't know. He just it was an interesting observation. In fact, it took about 10 years from the time he made that observation before the first treatment was ever administered. And the lesson in science on this is that it's really good for us to be building out our networks with people that have expertise outside of what it is that we do, because if Fleming had invested in those relationships, very likely he would have been able to reach out to maybe a chemist or another, biologist, and been able to share that observation with them, and they would have noticed it sooner and would have saved even more lives.
Yeah.
This is another photo of Fleming, and this is the office that he used to work out of. And I think it's funny, like, even though this was like almost 100 years ago, I think it looks very similar to a lot of our lives.
It looks so similar to my office. It looks so much like mine. Oh, yeah. I'm a cluttered mess, just like that.
Well, and the picture of you, Josh, on your website, like you've got a sterilizer in the background, and like you're just sitting there grinning ear to ear, and there's all sorts of petri dish plates and all this other stuff too. It's awesome.
Yeah. I mean, science is so fun, especially if you can have the freedom to be like, I don't have necessarily like an objective. I'm doing it. Maybe I'll discover something. But even look at Fleming. I mean, and so many of the great discoveries in science have happened by accident. It's sort of just like positioning yourself to be ready to be looking for that happy little accident to happen.
Well, that's actually how I got into mycology, right?
No, tell me. I'm going to stop sharing so I can see.
So I was in college, and I was in my first year. We were just my first it was biodiversity 101. It's the biodiversity was in that school, the biologist biology class, right? So we were doing all kinds of stuff. Like, that's where I learned to do a lot of the swabs and, hunting around for different things. But we were doing sea fern experiments. So just, to help us learn about genetics. So we're sitting there working with sea ferns, and one of my dishes got contaminated with mold. And we were observing the sea ferns under the microscope, and I just happened to see this mold growing. And to this day, I wish I knew what it was, but it was growing, and I literally saw these like tips kind of curve in on themselves, start to swell with this like bulbous end on them, and all of a sudden they just turned black, like something was injected into it, and then it would literally just like flick it. And the first time I saw that, I went, oh my God, it flicked it. It freaking flicked it. And my professor came over and she's like, what's going on? You know, because of course my outburst had interrupted the class. And I was like, it flicked it. It freaking flicked it. I was at a loss for words. And so she just looks under the microscope and she went, huh, okay. So, that's a little, what is it? Oh my God, it freaking flicked it. You know, she was like, wow. And I looked at her, and we had conversations earlier. She wanted me to change my majors anyways. And I was like, I'm thinking about changing my major to mycology. And she said, I think that is one of the best ideas ever. She's like, mycology, she said, it's about to change the world over the next 50 years. She said so this was like 2009 maybe. And she's like, I think mycology is going to change the world. It's going to touch everything from fiber, fuel, medicine. It's going to material science. It's going to touch everything. I was like, okay, that sounds good. That's what I'll do. And then I ended up throughout the rest of the semester growing mushrooms at home and then selling them, and then going, I'm stacking up student loans, but I'm making money growing mushrooms. I think I'll just grow mushrooms. So I quit school, and here I am. That's how I got started.
Yeah, and I was a little jealous because I was in the humanities and art department taking not a biologist biology class and like dissecting sheep eyes and stuff. And whenever he mentioned that, I was like, oh man, yours sounds so much cooler than what I
oh my God, that class was cool. For one thing, my professor in particular, because it was just a community college, and I mean, I don't mean to crap on community colleges because I think they're valuable, but it's not what you think of when you're thinking of like, oh, I'm going to get the best biology classes ever. But she was the head of the Knoxville Zoo for a long time. So like we took a few field trips there. Like we were constantly in the field, doing different surveys and whatnot. But like when we got to go to the zoo, like I got to work with the elephants one day, and I got to work with, such and such and such and such. And so we got to go to the zoo with this ornithologist who got out of his the vehicle that she drove him up, and he got out of the vehicle and puts these glasses on and then flicks out a stick and starts going across. And I'm like, so I started talking to this guy, and I'm like, how does an ornithologist like, how do you do it when you're blind? Like, so you can't see the birds. And he went, you hear that? I was like, yeah. And he's like, it's that kind of bird. You hear that? It's that kind of bird. And you hear that? It's that kind of bird. And he's like, apparently legitimately collected bird sounds and had like the world's largest sample library of different bird sounds from all over the world. And just, that's what he did, was he studied the sound and stuff. And I was like, man, anybody can really contribute to this stuff. Like, it's crazy.
Can you imagine, Josh, how many samples you could possibly get out of elephant poo? Or swapping the feet of the birds that are caught and edited and studied and released again.
Oh, I can get you all kinds of exotic animal poo if that's what you're into.
You made me an offer. I made an offer to your son, your oldest son, yep. I think. I was like, hey, if you can get me some of that bat guano to sample, I'll make it worth your while. They're
supposed to be coming back soon. I know he's planning on doing it, but they're not here right now. So, he did finally find a migration home or whatever. So when they get here, he's sourced it. He found it last winter or whatever. So now we're just waiting for the bats to come back. But we're totally gonna get you. But just depends on what kind of poo you want, man.
Yeah, he hiked for days trying to find a cave.
Yeah, he looked and looked. Come to find out, there was one right off the road, so you can just drive right up to it. But we didn't know about it back then.
Yeah, you were mentioning about the number of fungal species. Part of why we know there's so many species, they do this thing called meta genome barcoding, where you can take like a sample of soil and you can collect and sequence all these sequences of these, partial sequences of different organisms. And so we're able to detect like hundreds of thousands of species of fungi. Most of them, though, have not been able to be cultured artificially in a lab. And it's suspected that most of the species of fungi are not discovered yet, partially because we haven't figured out how to culture them.
Interesting.
Just like there's certain mushrooms you can't cultivate them artificially because they require,
a web.
They require a host, or they require other microorganisms to actually, we don't even fully understand why we can't cultivate them all. But the same thing happens with microbes. There are a lot of microbes that need other microbes to survive. And studying that, it's called the consortium effect. Studying the consortium effect of microbes is kind of an emerging area of study for anybody that wants to really dig deep into the science and stuff.
Are you familiar with Jasmine Richardson?
I'm not.
Okay, she's like on Facebook in the mushroom growing groups and stuff, but she's pretty quiet these days. But she has been doing research into truffles, and she is finding that she's having far more success treating them with a host of organisms rather than just trying to culture pure truffle. She's got, I can't remember the number that she said, but there's some number of species that kind of just coexist, and actually the truffles thrive better when they have that, quote unquote, what we would think of as contaminant involved with it. So, that was probably the first time I'd really kind of heard of the consortium effect. Was she was discussing that?
Well, truffles are a great example. Most of them are mycorrhizal species anyways, and require a plant or a host like a tree. I've never seen it in person. I've watched a great documentary on a truffle farm, and it takes about, I think, 7 to 9 years to get your first truffle. So it's really a rich man's game, because you buy some land and you plant it with a bunch of trees, and then you inoculate it, and then the whole 7 to 9 years the guy was training his dog to sniff out the truffles, and then sure enough, come year 7, 8, 9, they got their first batch of truffles and started harvesting them. And you can make some good money doing that, but it's a sizable investment and a huge amount of risk. I mean, imagine investing that much up front and being like, I hope in 9 years truffles grow.
Well, yeah, and then that is the thing, is that it's a hope. I mean, from my understanding now, we had Dr. Oman here at one point, and he's done work with truffles, and apparently you can get pretty decent at having a success rate, but like you're talking about, it's almost a decade before you have that success. So, and even then, the success rate is probably better than 50%, but not better than 80. So, it's still, like you're talking about, a lot of money to be invested. A lot of people apparently plant them in the wrong climate and have all kinds of there's truffle farms here in Tennessee. There's a couple that were planted some 20 years ago or so, and it's not the highest value truffles, from my understanding. It's, they planted some and then ended up with a Tennessee native truffle of some type, and that's the one that they actually grow and sell, because the Paragord or whatever that they tried to plant did not do so well. So and who knows why? Like, it's the wrong soil, wrong climate, wrong microbiome. Who knows?
Now, are those parasitic or symbiotic?
They're symbiotic, as far as I know. Truffles. I think that it's a true mycorrhizal pairing versus a parasitic relationship. But I'm not actually sure.
They're exchanging different sugars and carbohydrates between the roots and the fungus.
Yeah, that's my understanding too. Is that they the truffles probably like mine minerals and things like that and exchange it to the plant host for like he's saying, sugars and carbohydrates, because plants are wonderful sugar producers. So, interestingly enough, going back to the penicillin, one of the things that was mentioned was that the United States had a huge advantage in penicillin production, in part because we're corn growers, because corn is a native grain here, and it's grown to such extent. And apparently that corn dextrose was exactly what the penicillin needed to produce. So you were talking about like different feedstocks and things producing the enzymes that we need. Apparently corn was pretty crucial in that. And so like Germany actually had antibiotic production as well, but they had different shortages and stuff just constantly, but they also they did not have the kind of grain sugar that made it the the best way to go. But because of all the bourbon production in the United States and the moonshine production, they had plenty of corn sugar.
I previously worked for a very large biotech company that did large-scale fermentation of genetically engineered yeast. And the way that they made it economical was actually buying a huge amount of land in South America where they grew sugar, cane sugar. And then they built a huge biomanufacturing facility down there as well, because you want to have your fermentation tanks basically right on the farm, right next to all of these sugar canes. And so they would feed the yeast these sugar cane and produce the genetically engineered product at the end of the day.
Interesting.
Yeah.
What were they producing?
Let's see if I can say it without giving away the name of the company. It's a cosmetic compound that typically would come from shark liver oils. So you'd actually have to harvest sharks and then harvest their livers to get this compound.
And so with this, they don't have to.
It goes in high-end cosmetics. Well, it turns out you can figure out what the genes are for that to produce that compound, put them in Saccharomyces cerevisiae, which is your standard household yeast. And instead of producing alcohol, you'll produce this compound instead.
Interesting. I'm familiar with some of that, as in like the vanilla production being done through a yeast, or maybe that one was through an E. coli. Golly, I can't remember. I know that there are companies trying to produce THC and psilocin and things like that through different modifications to yeast and things. But I mean, there's apparently a whole bunch of stuff that they're doing that way. So
and that's also kind of what people when people look at molds, for example, and they find a new compound, they're not just interested in that compound. I mean, they could certainly grow it out, but they'll most likely end up sequencing the full organism and trying to find the genes involved with that particular compound. Because once they have the genes, then they can start. The industry term is chassis. They could start trying different chassis, whether it's a certain species of yeast or E. coli. They can put those genes into those organisms and see if those chassis will be better producers of that compound.
I like it. So it's like a car body. You're just swapping engines or something. This is what they're trying to do. They're trying to say with that. Oh goodness. Well, that reminds me. There was that. Was it one of the ones I sent you? The Felina Amphicordae, something like that.
That would be. That's one I've isolated on coyote poop before.
Is it? Okay. The one that had the big long white spires? Is that the...
Oh, yeah. I had another person on Instagram. I can't, I hope he's not listening. I can't remember his name, but he also sent me a sample with some of that growing. But yeah, that was a really cool Felina Amphicorda. We could pull that observation of it. It'd be kind of cool.
Oh, sure.
Some cool photos.
Where do we go to pull that up? Or are you pulling it up?
Yeah, give me a second to look for it.
Okay.
I'll find it.
Well, so somebody had actually sent that one to me, and I think he isolated it off. So it was a contamination in his mushroom op. He was having it in everything. So his laboratory, like all of his plates, everything was constantly getting it. And he's like, I don't understand. And then so he sent me a sample, and I'm like, I don't know what to do with this. Like, this is something I have never. This is alien looking. So that's why I sent it to you, right? Because I was like, Josh knows what to do with weird stuff.
Someone in another discipline, like you mentioned earlier.
Exactly. Exactly. There we go. Like, I've got what Fleming doesn't, a network. But come to find out, he had. Oh, here we go. Bam! Look at that weirdness, people. But he had found the cause shortly after I sent the sample to you. His cats were defecating between the walls of his laboratory and something else, and they had decided to use that as a litter box, and that stuff was growing all in it. So that is such a crazy looking. So is there anything special about that one?
I'd have to go check the literature. I mean, a lot of these, because there's so many species, a lot of them just haven't been studied. That's the exciting part.
Okay.
If someone could grow this out in liquid culture and do an extract and then put it under a mass spec or HPLC and determine, wow, this thing produces a powerful antioxidant that we didn't know about. And that's kind of the opportunity that I think that exists for mold. See, you mentioned that it came from a cat, and that's probably where it got its name.
Felina.
Yeah, feline. And it's probably also why it's commonly associated with different dung from different. I mean, I did one from a coyote, which is not quite a cat, but
yeah,
it's somewhat associated.
It's probably where the coyote ate someone's cat, right? And then
gosh.
Actually, you might not be wrong about that. Actually.
Well, and honestly, we probably ought to audit that name because it said by Andrew Reed.
It said Woody at the bottom too.
Okay, good. Because I credit where credit's due, in my opinion.
Yeah, Woody's the one that sent that to me. So.
Also,
well, you sent it to me, so I said submitted by you, but yeah.
No. I mean, he would have never submitted it to you if I hadn't submitted it. So we'll call it good.
Collaboration. But can you imagine being sick and then turning around and your doctor's like, "Hey, so there's this, really nice antibiotic that would save your life, but it was isolated from cat poop."
Oh, I wouldn't care.
I wouldn't either. I mean, at that point, it's clear it's sterile culture, so you're not getting getting all the germs.
Yeah, and if you ever go and take like my wife takes these supplements that has like has a bunch of bacteria, like healthy gut bacteria,
Probiotics.
Yeah, exactly. I mean, and that stuff is commonly found in human poop, so it's like you just have to isolate it, and then it's the organism by itself. It's not. A good portion of the human poop is bacteria, dead bacteria. So
that's true. And with 3 children who have somehow or another continuously managed to land themselves in the emergency room, I've heard some really strange conversations, and that was actually one of them where they transferred human poop from one person who was considered very, very, very not abnormally, but very healthy, and then they inserted it into someone else that was struggling, and that was a very strange medical procedure, in my opinion.
South Park did a whole
fecal transplant.
Yep, South Park did a whole episode on it where everyone's going after Tom Brady's, like it's the spice. So someone here,
they don't last because a lot of people have they're missing a gene, or they have some issue with their genetics that inhibits that bacteria to stay alive. So I mean, it's sort of like you could do a fecal transplant or just take the probiotics,
Well, you can take the probiotics, but at the same time, like you're saying, you can only host what you can host. So, and part of that's your diet, I'm sure. They've actually, I saw a breakdown where many Americans have far more fungus in our guts than a lot of people around the world do. And they think that's because we have a higher than average, carbohydrate intake versus fat intake, where a lot of other people in the world do. So there's that. But then, I was reading recently about some gene expression mutations or whatever that cause you to produce certain enzymes that will support different bacteria or whatnot. So it's
in all seriousness, it is very important work.
Oh, sure, definitely.
Yeah. Real quick, Oz TV says Anatolian savanna is full of oaks and truffles. He was going after the one below it. High copy plasmid with ADH promoter. You can overexpress many genes in yeast. However, transforming fungal cells are not easy because making protoplasm with mycelium is not easy, necessary for transformation. Agrobacterium method works better. So I don't know anything about that stuff right now. I understood the words, but I did not understand the meaning.
Conceptually, that's not incorrect. That's not accurate conceptually. Practically speaking, agrobacteria is great for prototyping. Industrially, it's not used very much because you actually have to transform 2 organisms. You have to first transform a species of bacteria called Agrobacteria tumefaciens. So you put your gene in the agrobacteria, and then you use the bacteria. The Agrobacteria tumefaciens is what causes tumors on trees and plants. If you've ever seen a tree, sometimes it looks like when you have a tree and you prune it, and it forms like a knot over where you cut the limb off.
Yeah.
You know what I'm saying? When you ever see a big nodule on a tree,
Oh,
sometimes those are caused by agrobacterial tumefaciens, which is bacterial infection. It has the ability to transfer DNA from the bacteria to the host organism. Scientists have figured out how to hijack that technique to transfect fungal cells. So they'll put the gene or the plasmid that they want to put into the fungus. They'll put that first into the bacteria, and then they'll basically put that agrobacteria, mix it with fungal cells, and the agrobacteria will transfect the plasmid gene into the fungal cells, which is pretty cool.
That is pretty cool.
But practically speaking, that's it's very finicky. So industrially at a commercial scale, there's a hesitation to use that. That's why traditionally, like right now, it's hard to get people to use new fungal chassis. Use that word again, chassis. They typically want to use something like yeast or E. coli because they're so much easier to transform. But I think that'll change. I think there's, I think fungal transformation, genetically modifying fungus, is still pretty nascent, and we need new technologies. And there's several techniques you can use to transform them. But that's pretty accurate. It's pretty spot on.
I know the Odin project was discussing some of this. They were talking about the difficulties in genetically modifying fungus in general. Like apparently, one aspect is that it's really difficult to basically get past the cell wall without destroying what's on the inside. Just because of the very nature of chitin, being so tough, and it's so difficult to break open.
Almost all fungal transformations will start with mixing the fungal cells with a detergent to kind of get that chitin wall off and expose the protoplasm. And then there's just, then it's a matter of, well, what technique do you want to use to physically get a piece of DNA inside that cell without, killing the cell? Yeah. But then I think the bigger issue is with fungus especially, it's hard to get those changes to stick around. So you can get the fungus, you can get the genes inside that fungus, and it might replicate with those genes a couple of times, but it very, very often will start replicating and no longer express those genes.
Interesting.
Wow.
Yeah,
that makes me think. I wonder in part if that's like, if I'm working with the material, the mycelium. If I can get the gene into a few cells, that's all well and good. But if the other cells are not modified and they're continuing to grow, then is it something like that, or is it just that the actual DNA replication machinery can recognize, oh, this doesn't belong, and then work its way out?
That's right. And not to bring up a hot button topic, but mRNA vaccines kind of rely on this, right? So when you get an mRNA vaccine injected into your arm, there's a small piece of DNA that's going to be injected literally inside of your muscle cell. A few of those muscle cells that receive that DNA will recognize it and will actually transcribe it and will produce a protein. Your body is going to recognize that protein and start producing antigens for that protein. That protein happens to be the vaccine protein. And so it just doesn't have the viral part of it. It just has the protein that is attached to the virus. And then your body will produce these antigens, and that's what builds the immunity. But very shortly after that happens, the cells are replicating, and eventually they're going to stop producing that protein and stop replicating that mRNA. And then all the previous cells that were producing it die off and get excreted. And then you no longer have that exogenous DNA in your body anymore, but you have the antibodies that were produced from the protein that was produced by the cells that got injected with the mRNA.
Interesting. Is that hence the boosters?
Yeah, well, the boosters are often because that protein mutates. And so every time that protein mutates, you need a new gene sequence to produce a new version of that protein, so your body will produce a new antibody in response to that new protein. Okay. Interesting. And same thing with like the flu vaccine, it's typically only 50% effective, just because it evolves so quickly every year. And, we'd be literally, if we wanted to go get a vaccine every day for something with all the mutations, we probably could. Not, I wouldn't advise it, and it's probably not practical. But the reasoning is why is because, those mRNAs are very specific to a very, very specific protein that they're trying to emulate. And if the virus in the wild mutates even just very subtly, you've got a new shaped protein and a new mRNA that needs to be generated.
Yeah. What are they called on the outside of the spike proteins? Is that?
Yeah, exactly. The spike protein is kind of attached to the viral payload, and that's what's drawn to your respiratory system. So they're basically having you produce that spike protein without the virus, so that your body will develop an immunity to that spike protein, so that next time you come in contact with the virus in the wild that has that spike protein, your body already has immunity to it. It can recognize it and be like, no, we've seen you before. We're not going to let you attach.
I know you.
Yeah,
exactly. You're not getting through here. Well, I'll tell you what, Josh, we're an hour in. I need to use the restroom. So I was going to do a quick word with our sponsors. So if you don't mind, we'll be back in just a minute and a half, 2 minutes.
Yeah, sounds good to me.
Awesome. Be right back. All right, guys, and a word from our sponsors.
Introducing the future of mycology, Dr. Silurian's Elixir of Mycelial Mastery. This isn't your average culture mix. It's a revolution in a packet, crafted for the dedicated cultivator seeking unparalleled growth. Experience the visual alchemy of this masterful brew. As you mix 10 grams with 500 milliliters of water, watch the solution transform into a rich, amber spectacle, the hallmark of a nutrient-dense sanctuary for your burgeoning mycelium. This is where vibrant color meets vibrant life, setting our elixir apart from the ordinary clear liquid cultures. Prepare to be captivated. Mix 10 grams of Dr. Silurian's elixir with 500 milliliters of water, add a stir bar for thorough integration, and sterilize the blend at 15 psi for 45 minutes. Once cooled, introduce your favorite culture into this perfectly prepared, nutrient-rich environment. Watch in awe as the elixir works its magic, unveiling a mycelial masterpiece before your eyes. Choose Dr. Silurian's Elixir of Mycelial Mastery for a mycological adventure that's as visually enchanting as it is potent.
All right, folks, get you some of that. Also, really quick, I'm going to bring Josh back here as well, but when you go get you some of that liquid culture premix, you save yourself 20% on our website. It's any product, any service, 20% off with I Love Mold 20. So that was Josh's idea. So we know as mushroom growers, you don't typically love mold, but today you will with I Love Mold 20. That's ILUVMOLED MOLD 20. So awesome. Welcome back, Josh.
Molded.
Hey guys.
I got to do my, I got to make my bread. So
I get it. Totally cool.
plus,
Actually.
Samantha feeds me a lot of coffee. So.
Oh, okay.
Do what, baby?
Well, just the discount, it helps other people make their bread, really. It kind of, it takes a little bit away from ours, but that's
right.
fine. We want people to be able to use it and make money off of it, too.
Absolutely. All right.
Funding.
Let's see. We have Powerman says, question, I can give you some samples of fungus that have proven to be resistant to most medication. Maybe you can come up with a mold that has an effect on it. He said, I look for odd fungal samples, not necessarily mushroom, to see what characteristics it has. Are you taking samples?
Under very specific criteria. Otherwise, I get inundated often. I literally have people in my town that will drop samples off on my front porch daily. So,
you say daily?
I do have to be a little bit discretionary on what I take, because I've looked at so many molds that a lot of times I can just tell by sight what the genus is.
Yeah.
My channel has not been updated. I used to have hair in some of those videos, so that tells you how long ago it was.
I was gonna say that I know that I have shared stuff with you. I'm like, hey, do you want this? And you're like, that's a common thing.
Yeah, and I think it's cool. I just have to say, like, I can't take everything, or I'll be like. It took me a year, by the way, to process those bioprospecting samples from my visits to you guys.
Oh, yeah. I want to talk to you about that, actually.
Yeah.
Is now a good time to bring that up? Yeah.
Yeah. Okay. Well, you want to talk about this pot? You want to talk about these time lapses before we get into that?
Let's talk about the time lapses first. You're right. I'm too ADHD, man. I appreciate you keeping me on track.
I forgot about this one. I'm bummed. It's been up for a year. It only has 400 views. That's
people go check out EverymanBio on YouTube and subscribe. He hasn't done much in the past little bit, but you should go check out the time lapses. If we get him some subscribers, maybe he'll do some more. I was that one right there, the Xylaria. Let me pull that one up real quick because that one I watched last night as well. And Nito. It's going to have the inhibition zone that we were just talking about as well.
This one has a really cool story associated with it.
Yeah, go ahead, tell it.
Yeah, I'm like entranced by the time lapse. I'm like, wait a second, let me hit the edge. It's like that old screensaver where the ball bounces around and you hope that it hits the corner. And I'm like, will it touch?
I know exactly what you're talking about.
I gotta see if it's gonna touch. All right. This wonderful. See, I can't even talk while looking at it. It's so satisfying. This mycologist from the University of North Carolina, he reached out to me. Jose Varaja, Dr. Jose Varaja, who, by the way, helped put together some of the most awesome DNA barcoding protocols that are being used by the amateur community. He's like, hey, we're studying these two species. We found this anti-cancer compound that only occurs at the intersection of this Xylaria flabiliformis and this Aspergillus fisheri.
Really?
Yeah. So, and they built this really cool robotic tool too, that when those two fungus grow together, they have this little probe that will robotically go and follow it and stick the probe right at the intersection. So if you pause the video, but then I want you to I'll tell you where to pause. Pause it when they're touching and there's a little bit of an overlap between the two. You'll see like an intense white ring right around there. Let it go like a half a second more. Right there. Stop.
Yeah, that's almost good. Like it hits, bounces back.
If you look at what you call the margin on the right, on like in the middle, where they're touching, it's a little bit whiter there. That's kind of where the two are touching. That's a probe that will go right into that particular section, and that compound is only produced when those two are in contact with each other. And it's believed to be, a defense mechanism from one or the other organism, from a competing organism. And this is a great example of what I would say is like that consortium effect, which is really hard to study because it's hard enough to go out, find molds and study all the compounds they could produce. But then if you go, well, what happens if we start studying all the compounds produced when molds come in contact with each other? That exponentially grows in size. So they said, hey, would you be up for, we're going to present our paper on this. Would you be up for shooting a time lapse and letting us show it? And I was like, sure. So that was a really great example. This ended up being a contaminant from which I thought was kind of cool. It was unintentional, but
goodness, watch that one just sweep the field.
Yeah, and it just really shows you the power of Aspergillus, which is the black one that's overtaking everything. It produces some really powerful enzymes, but even make it hard for the enzymes involved in DNA barcoding to work sometimes.
Interesting. I've familiar with the fact that Koji is an Aspergillus.
Yes.
Which is interesting because a lot of Aspergillus is actually not pretty harmful to us, right? Like,
well, a lot of molds, if you are immunocompromised, can impact you. Look, soil has all sorts of bacteria in it that can kill you, right? And the same thing with mold flying around in the air. if you're immunocompromised and you overwhelm your system, you can get an infection. And so there is aspergillosis, and you can get a pretty nasty infection from that. But we have a natural immune system that protects us from all of these different microbes, viruses, and fungi, and bacteria. So that's the answer I always give, is like, there are some molds, just like there are some bacteria and viruses that are more infectious than others, and you should exercise caution. And we can talk about some of the things you can do. A lot of it, most of what I would have to say is common sense. Don't open a plate and hit it and then inhale.
Have you seen the show? Have you seen the show?
Exactly. What's that?
Have you seen the show Hot Zone?
No, I haven't.
It's really good. I really, really enjoyed it. The first season is about the Ebola outbreak in the 1980s. And then the second one is about the anthrax scare from back whenever you and I were kids.
I remember watching that one.
Yep. But it's a good show.
I saw one about the anthrax, about the guy that he was basically doing a liquid culture. That's how he produced the anthrax that he put in envelopes. He was doing a liquid culture of anthrax.
Liquid culture, and then just drying it until he had the powder, and then using it's crazy. Like
and anthrax is a
little bit, Josh.
field.
Do what?
What's that?
I said that actually scares me a little bit. Where it's like, oh man, I can do this. I can do this in my basement. The other kids are doing this.
We're not going to.
No, I'm not going to.
That sounds like my wife. She shuts it down before the thought is even allowed to.
Well, she knows that I'm a mad scientist at heart. So I literally have. It's called Andrew's Death Box. And it sits under one of my shelves, and it's like every poisonous mushroom that I've ever come across so I've got like spore prints or clones of it, but it stays locked up and away so that it is never mixed into the general population. All my weird molds and spore swabs that I've taken of molds and things like that are all in there. So.
Yeah, I mean, if you get a hold of enough cherry pits, you could also easily isolate a bunch of cyanide. So.
I may not work on that one.
I think all of this stuff is in the Anarchist Cookbook, if you're familiar with that.
Oh, I've never actually read it, but I've heard of it. You could say it's pretty easy to download these days, but
yeah, it is so easy. And nowadays, like if you read it, it's like, oh, this is tame. Yeah,
I imagine so compared to the stuff we see nowadays. Like, oh my gosh,
exactly.
I bring up the hot zone because that's actually something. One of the guys is like, oh, it's not anything dangerous. It's like simian whatever virus, which is non-harmful to humans. And he's like, your samples are just messed up. So he starts culturing it out, and he shakes and then smells the flask, and then has his lab partner do it. And this happened in real life because if it's was it called Pseudomonas? Is that the common soil bacteria, Pseudomonas?
That's common bacteria.
Apparently, it gives off a grapefruit smell. So he's like, "Oh, check it out! It's smell it." And he's like, "Why?" And he's like, "If it's grapefruit, it's going to be Pseudomonas." And they couldn't smell anything. They're like, "Oh, that's crazy." And then later on, they find out, "Oh, this variant of Ebola is airborne." So they both sniffed the flask where he had cultured Ebola, and it was like he took no freaking precautions whatsoever. And he was working for U.S. What's it called? The U.S. AMRID. I think it's what it's called. It's the United States Biology Defense Program. I can't remember what it's called now, but
you have Google.
Right. Back to the time lapses. How do you keep yours from? How do you keep them from fogging up? Because that's crazy to me. How you're able to do that?
Yeah, so when you're shooting the time lapse, when you've got, I shoot them in I have this cheap Amazon picture box. I want to talk about this one. This is actually really cool. Let me talk about this one.
Yeah, talk about this.
It's all his babies.
So the plug in the middle is a plug of Copernellus mushroom, which is a very common mushroom.
Mm-hmm.
It grows on logs and whatnot. And when I was trying to isolate some stuff from a log, and I noticed that there was a bunch of stuff. There was a dilution plate, and there was a bunch of stuff growing. And I noticed that there was one section of the plate that was producing purple. And I was like, well, when you don't see blues and purples very often in nature. So if you do see them, pay attention because they're kind of rare. And so I isolated the copernose by itself, and it didn't produce the purple. So I had a hunch that it had something to do with it only being produced when it's under some sort of stress. So I said, well, wouldn't it be cool if I took like a sterile swab of penicillin, and I dipped it in a spore solution, and I just lightly swabbed the outside of the plate, and I put a plug of the copernose in the middle, so that as the copernose grew out, it would sort of come in contact with the penicillium. And then I photographed the time lapse. So you can kind of see here the penicillium is growing, the copernose is growing from the right. And then as they meet up, in defense, the copernose produces this compound to protect itself from being overtaken by the penicillium. And that's what the purple is.
Interesting.
Which is really, really cool. And that has not been characterized yet. So that's something that's new to science. No one has, someone has to be interested and be willing to put in the time and energy and money to research what that compound is. So, if someone's listening and has access to a lab and wants to co-author a paper with me on class or characterizing that compound, that'd be really cool. So that's the story with that. The short answer is with the plates, you have to put something on the plate to prevent the fogging. So
okay.
A very poor man's technique is take some dish soap and lightly smear a very thin layer of dish soap over the underside of the lid while you're in front of a flow hood so you can keep your plate sterile.
That's
okay. Cool. Interesting.
So, because as it begins to want to fog up, there will be nothing for it to cling on to. The best solution though is what I have right here, which is you can buy anti-fog solutions on Amazon for your glasses. For people that go skiing, you spray it on the inside. You can use that. The best one I found is this blue gel Clearview spray. My background's not gonna work, but blue gel Clearview spray. This is for my gel electrophoresis box.
Yeah.
That's cool.
And it's a similar solution, but their formulation, whatever it is, it works great. And it just leaves a very, very thin film of some sort of oily surfactant, like a soap type material. And that, kind of prevents. The other thing is it doesn't work too well in hot environments. I've done it before where I've done a time lapse in a very warm environment, and it eventually, the condensation will just start to accumulate and drip.
Gotcha.
It doesn't matter how much you put on there. So best to try to keep it cool, which is hard to do because if you're shooting a time lapse, you need an external light source pointing at the thing, and then the heat from the light can create the condensation. But if you use LED lights, that was another way I've countered it, is just use an LED light, because LEDs put off virtually no heat. So
yeah, I tried to do through my microscope. I tried to do some time lapses, and it worked pretty well. But I mean, even with the LED light, stuff fogged up on me. Because I bought an aftermarket LED ring that goes on the bottom of the microscope, and that ended up putting out enough heat that it caused plenty of fogging. But I can use. I'll definitely try an anti-fog. So, are you putting parafilm around your plates while you're shooting them, or no? I am.
So another trick that I use is instead of taking the parafilm and doubling it up, like folding it in half.
Mm-hmm.
I don't know how you do yours, but that's
what we do.
Fold it in half. I actually will cut it in half, so it's actually much thinner than you would normally use. Mm-hmm. And then I won't stretch it all. I'll leave like half an inch at the top open, because part of a reason why it also condensates, there's not enough air exchange. So if it's hot, the pressure changes. So I put enough paraffin on there, a very thin layer, so that it doesn't dry out while I'm filming it. but I also leave a small gap at the, somewhere along the edge so that there's air exchange.
Okay. I will try it then. I'll definitely try that again because I was thinking like, okay, if I set the camera and the thing up on a timer where the light just strobes.
And I have a timer set up, and I take a photo every 20 minutes. And I have used the same camera for 5 years, so I have not ruined a camera. And it's a DSLR Canon, so I don't know what you're doing with your cameras, but like
about a picture every 10 seconds.
That will maybe do it. But obviously, the more photos you take, the smoother it is.
Yeah.
But I've molds grow so quickly. That also plays a big role in it. Molds grow so quickly that I don't need as many photos. Like one every 10 to 20 minutes is pretty good.
Gotcha. Yeah. See, mine will be going picture every 10 seconds for days while I'm capturing time. Because macro fungus that's the problem, right? Is that mold and bacteria beat us out if they're there because of the speed aspect. Like if we can overwhelm it and get enough biomass in there, then sometimes we can compete. But
that Copernellus time lapse you just watched, Copernellus is a mushroom, so that probably took 2 to 3 weeks to shoot for just a minute of time lapse. Okay, you're talking 3,000 to 4,000 photos. So it's just a lot. It is a lot of work. That's kind of why
I think my average was about 20,000 photos per time lapse I took. So yeah,
and it's like you've got to stitch them all together, and then I like to edit. To create a compelling video, you don't want to just show like here's a time lapse end to end. You want to zoom in some areas. You want to like reverse it and just kind of play with it a little bit, make it more engaging.
Oh yeah, Samantha's wanting to show you something. I can tell. So
yeah, please.
Let's see. There we go. I'll biggie us a little bit so she can show it off there. But all right, Samantha, you got the con.
So we'll make like 300 bags, and then maybe 1 or 2 of them will be contaminated at some point. And something that whenever you mention the coloring, I've noticed that some of the mushrooms will make kind of colored metabolite. I'm not sure if it's from this one. It's kind of a maroon, almost orangey kind of color. I'm not sure if you can really see. I didn't know what you thought about that.
There we go. Or there's a good pocket of it.
Here, let me unmute myself so I can hear his response.
I can see that. Yeah, it looks. Yeah, I could definitely see kind of around the borders how it's brown.
Mm-hmm. And then
where it meets.
Yeah, it's got a couple of pockets of just the juice, but it's definitely an odd color. Like compared, like I'm used to seeing really yellow, or amber color, but this one the camera's not really quite grabbing it. But it's like almost got a maroon tint to it, or almost like a purplish tint to it. I don't know. It's an odd metabolite. That's from a shiitake though.
And normally I would see a lipstick mold that color, but it's not as bright, like a magenta, pink, red kind of as a lipstick mold, but it's definitely something else.
Is there green there in between the brown part?
Yep.
So it definitely has a mold.
Oh yeah.
That's interesting.
It's like seafoam green. It's really light.
Yeah, that's most likely a penicillium. There's over 500 species of penicillium.
Yeah, looking at that, I was like, okay, that's penicillin most likely because the trichoderma tends to be like a much more of like a aquamarine or something like that. Like I don't know the color exactly, but it's definitely a different kind of green for trichoderma.
And I have these nodes on the side. Can you see this?
Sometimes you can tell by the
popcorning.
Okay. That's
just where the bag's popcorning. What'd you say, Josh?
Sometimes you can tell by the texture of the molds to differentiate between penicillin and trichoderma. But honestly, I've been tricked by them. They can be quite similar, and both of them are huge genera. They both contain hundreds and hundreds of species.
I noticed because I was looking into a buddy of mine started a plant care company, and I was looking at different trichodermas to do that. And you could order all kinds of different trichodermas, but for the most part, it's all a hybrid species that they have patented. So they like almost every single one of them that I saw that was commercially available. Like if I just wanted Trichoderma aggressivum, one of the most common ones, it was nowhere. Huh?
Aggressivum?
Oh, I mean, it's right. It's super. Yeah, I mean, that's the name was aptly put, but it was like you couldn't find it. You could not find it. Now, granted, it's supposed to be one of the most common soil-borne ones, so if I wanted, I could probably find it. But it was just interesting because everything they offered was a hybrid between two Trichoderma species. And I was like, how are I was like, dang, they've got a ton of these. Like, how in the world are they hybridizing so many? But
that's interesting. I haven't heard of that. That's cool.
Yeah, it was pretty crazy looking. I was also looking at our dog got fleas because our cats have now decided they're indoor-outdoor cats, which is about the time. But when they brought fleas back in, I was like, oh, we could get all of this stuff, and we treated him like we normally would. But it got me thinking, man, I want to start looking for something that'll kill fleas. And I found out there are molds that will attack the instar phase and like different phases of fleas, and I was like, oh man, I might order that. So then I started like finding all these like companies that just have libraries of different, there are different molds and stuff for sale. They all want you to contact them and get a quote. They won't just sell it to you the way I sell a strain off a website or something. But
what if Teddy's allergic to mold?
You can get some Bavaria bassiana, which is which is an insect associated, and it's approved for sale. And it's safe for use in agriculture, and people will use that for people use it for bed bug treatment, just general pest control. So it's called Bavaria bassiana. Yeah, a subsection of my collection are strains that I've isolated from insects, because that's an area of research that people are really interested in for biological pest control. So that's one thing where if someone goes, hey, I found this bug that's infected. Are you interested? I'm almost always interested in that. The question is, are you going to be able to get it to me before it dies? And that's the harder problem.
Yeah. Yeah, I can imagine. That's gotta be so crazy to me. I'm excited by the idea of like just buying a bag of spores and dusting my dog with spores and dusting all the cracks with spores, and there's like no fleas ever again.
I'm more interested in, can you collect enough of the fleas that you could put them in a small sterile tube, crush them, add some sterile water, and plate it? And you might find some really fascinating.
Oh man, if we had not, we treated him so all the fleas are dead. As far like we've not been able to find a single one on him for 2 weeks. So
and it would take some time to get enough together.
I would put my kids on it. My daughter would. My daughter would do it. I know she would.
Herd the cats.
Yeah, she would totally sit there and just grab the cats, and you'd see buttons.
If you're trying to do it, I'll give you the protocol of what you need to do to isolate the flea fungus.
That sounds good. I'll either do that or I'll send you a tube of fleas. So.
Love it. That would be a first, actually.
I saw a guy went to an oyster mushroom farm and swabbed the caps and stalks or stipes and stuff of oysters, and isolated a yeast, and then they brewed a beer with it, and then at a mushroom festival they served that oyster mushroom yeast beer. It was pretty good. So
cool.
So Niall Hippie Dad says, "Can you spell the insect killer strain in the comments, please?" Oh, you just did. Dang it, you already saw it. Dang it, man, you're on it. So there we go. Bavaria bassiana. That's a pretty name, actually.
And if you go on Amazon and type that in, then you'll find all these products selling the spores and liquid solutions for that.
Interesting.
And basically, Bavaria works because when the spores germinate, they drill into the to the host organism, like the insect.
I like it. That's molecular drilling, I think is what that's called.
Yeah.
It's terrifying.
It is kind of actually. It's pretty horrifying. Basically the hyphae use a combination of pressure, hydraulic pressure, and oxidizers to basically loosen the bonds, the molecular bonds, and then high pressure of the tip to just kind of find somewhere to wiggle in. And then it just does that repeatedly until it just drives through. It's super cool stuff.
Yeah,
it is.
Let's talk about you and me and bioprospecting and what the samples you process through. I'm super interested.
Yeah, do you want me to share those observations so we can look at them?
Yeah, please.
All right, let's do it.
I'm super excited because I don't think I've even seen everything that you've done. So we went to a bat cave, or not a bat cave, just a cave. We went to the grain basement that used to be, because this was a co-op for a really long time, and then the lime yard where you got a sample of the lime water pool. Let's see.
Yeah,
Okay,
so
let me pull you up
there's
here.
a lime quarry. We kind of broke the law and broke into a lime quarry and took a sample. So, sorry,
little bit.
quarry.
A little
Sorry,
gorilla bioprospecting never harmed anyone.
It was a small sample. It didn't hurt anybody. But this is a subset of those samples. There are a lot more, but these are the ones I'm prepared to show right now.
Okay.
There's a couple of really pretty ones that I want to show. So these two up here kind of jump out to me.
Yeah.
And let's just kind of take a high level look at these and look at the different what's called morphologies, the physical characteristics of these molds. We have some that look kind of flat and wrinkly.
Yeah.
And we have these fluffy ones, and then we have this really cool pithoascus. Ascus means stack in Greek. And so you can kind of see, we'll dig into this one.
Yeah.
But this produces little stacks of spores. You can see that. And then here are some that produce rings. And then you have the yeast. These are Basidiomycetes yeast, which is kind of cool. These are these pink and yellow yeast. Then we have here's a penicillium, a Cryptococcus neoformans. This can be pathogenic to humans. It's safe to work with, but this is just an example of one that you'll find in nature that's pathogenic. Polypocladium. This is one that commonly infects insects. Let's see. This cool Nemania. That's a yeast-like fungus.
That's beautiful.
Flat. It is very beautiful. We'll look at that one. This
Galaxies.
Oh, how do you say that one again?
I can't say that I'm pronouncing these correctly, but I would say this is Heliotialis, and this is at the order level because most likely it's a new species, most likely not described.
Oh, cool! You may have found a new species on our trip. That's neat.
Oh, I'm sure several of these, more than one. Like this, when things are not at the genus level, it was because there's typically not a species for it to place it in. So this one here looks like potentially a new species. Some of these, this Oniginalis possibly, it's at the order level. That means I don't even have a genus for it.
Oh wow!
Also, same with this Docetiale. So you see here how. Now, I only use one gene to DNA barcode these, and for a lot of molds, you need multiple genes. So it's possible with additional genes, but I start off with the universal barcode, which is called ITS. Generally speaking, if that doesn't even get you to genus level, there's a good chance it's just not described yet. So which one of those do you want to start at? Looking at?
Let's go to the top right one, the Docetialis.
Yeah, and these are, I just know from experience because I've isolated other Docetiales, and these are plant associated. They're commonly plant parasitic for plants. And when we got this from isolated for rotted sunflower seeds in a 100-year-old warehouse basement.
Yeah.
That's your warehouse basement. we went into the basement. You told me that it was 100 years old. It used to be like a sunflower seed. What's the story with that place?
Yeah, it used to be a farmer's co-op, and sometime before the Civil War, the railroad used to come through here, and so some point in the early 1900s, they built a warehouse right on the tracks. So our back docks are extended, but you can still see the old railroad bed that our back docks were built out into, where they used to just pull the train right up to the warehouse and unload right here. So it was before this place was here where it was supplied by rail rather than supplied by truck. So it was well before, that kind of infrastructure was really in place. And so it's just an old, I think that when I looked at the date later, it was built 1916.
Nice. Yeah, when I was doing this and doing some research, some of these ones that I found in the basement, other strains that were in the genetic database GenBank had been found in similar environments, which I thought was really, really cool. Agricultural basements, something to that effect. So it was like, cool. I knew I had the right specimen because other similar things had been found in those environments, which is cool. Like I have found like stuff here in caves that's only ever been found in another cave in the Ukraine, as an example.
Wow.
Which is pretty cool. So yeah, this is a pretty one. I love the features on this one, and extremely slow growing. Whenever you see all these wrinkles like this, then it puts so much energy in concentrating its growth in the center and not in outward expansion. Those tend to be very slow growing.
Kind of reminds me of that green one that I sent you.
The recent one?
Not the most recent one. I think I sent you one that was I don't remember what it was exactly, but it was some sort of green. I thought it was a mold, but I think you said it was a bacteria. But I could be wrong.
Oh yeah. I could. I vaguely recall it, but yeah, it's either a bacteria or a mycobacteria. Which there's several bacteria that can produce spores and even hyphae. But they're bacteria. They're not. So yeah, this is a really cool one though.
Yeah.
This is producing these tiny spore structures, these little balls, these little asci.
This kind of stuff really makes me want to like study this as like an artistic thing, and then like make a sci-fi movie where these are the landscapes, right? Like,
yeah,
it's just like literally a weird alien mountain.
It's just really cool. I mean, this stuff makes me so excited when I see it, and you never know what you're going to get. That's the other cool thing about working with molds. Like, I try to go for the weirdest samples, and even mundane stuff. You never know what's going to produce.
Right. Yeah.
This is a really pretty one. This is another one. I think we do have some lime quarry ones that we'll look at. This is kind of like has a felt-like appearance.
It looks super soft. Yeah, it looks like you could make a pillow with that. Well, what about that? Yeah, there we go.
So if you're to the untrained eye, you would think this might be a bacteria. It's got like a wet sort of surface morphology to it, and the colonies are round. You don't really see any hyphae, but this is a yeast. This is a fungus.
Yeah. To me, when I looked at it, I was like, "Oh, that looks like." To me, yeast always looks kind of. I don't know I hate to put it this way, but like greasier.
Yeah.
Than a lot of bacteria does.
It has a greasy look to it. It definitely has. Yeah, it can be. You can some of these photos make it look more shiny. But
so would that make a tasty beer, or is that a?
Well, actually, industrially, these red yeast, it's a category of yeast called red yeast, even though it doesn't look red here. It's kind of orange, but it's still in the red spectrum. They're known to produce biolipids, which are used in commercial manufacturing for like commercial surfactants or biodiesel. So this is in a class of organisms where they actually do produce lipids. So when you say it kind of looks greasy, that's not surprising. They are known for producing lipids. And that's how these are used. So just like a beer, just like how beer is brewed, you could put this in a huge vat with sugar and it will potentially produce a really interesting lipid that could be used industrially for something.
But probably not an alcohol.
Probably not an alcohol.
Ed Grand asks, are there any good online keys for mold identification?
No, because you can't identify mold through sight alone. I mean, look at some of these. Like, look at this Mortarella. I mean, I can identify genus level. I know when I'm seeing like a Mortarella or Penicillium glandicola is extremely common. Maybe not the glandicola, but the Penicillium. It's fairly easy to recognize. So I would say you can do genus by sight alone. Then you start needing a microscope to you can really pin down a genus with a microscope. But then you start running into Penicilliums under a microscope, which there's almost 600 species. Many of them look identical microscopically. So the only way to identify them is genetically, and even genetically, sometimes you need to sequence more than one gene to identify them. So,
isolated from creek sediment. So that's from the Mossy Creek area.
That's right. This is from Mossy Creek. Yes.
That's from the bog down there. Yeah.
Wait.
That
Mossy
is
Creek.
exactly what I would expect to come out of that bog. Let me tell you.
I just thought it was cool, these structures that it performed. This is a very mature specimen. You could, the plate's dried out, but you could see like as it grew, it produced these really cool shapes.
Yeah.
So I thought that was really, really neat.
What about that Cryptococcus?
Yeah, the Cryptococcus neoformis. This also came from your
And that's a human pathogen?
This is you could, somebody wants to look up the Wikipedia page for Cryptococcus neoformans.
That's a gram negative, right?
It's not bacteria, so there's no gram staining.
Okay. Interesting.
And almost all of these are going to be infectious to the. Now I'll tell you real quick. I can pull. I've got the Wikipedia page up for this. Yeah, it will infect. Most infections occur within the lungs as the fungus enters its host through the respiratory route. But you've got to be immunocompromised. So it's usually a secondary infection for AIDS patients who are immunocompromised.
Gotcha. I was gonna say my boys are the ones that just cleaned out that basement. So
I will say though, there are some of these like this one, which can be really difficult to treat if you do get infected. You have to inhale spores from this. So you look at it and you go, this thing produces spores, but if this thing dried out, I'm guessing, and you took a swab and you sniffed it real hard and you were not feeling too well, probably don't want to do that. We don't have a lot of
like a bio load that would be infectious even with a healthy immune system.
Yeah, something that your immune system can't keep up with it, right? So if your immune system can only, it's all chemistry at the end of the day. If you just overwhelm your immune system with infectious, microbes, it can only produce so much antibodies to those things. Only produce so much defense to that.
Yeah.
So,
Ed said I use the Illustrated Genera of Imperfect Fungi by H.L. Barnett. You familiar with that?
I believe so. It's a black and white photo.
That one, I don't know. I'm not familiar with it. It sounds cool. I did just get an illustrated guide to fungi that's got some beautiful photos of like mold and stuff, but it's like it's not really it's like a coffee table book, just different pictures. So
yeah,
I'll look at that one.
That's really cool.
See, are those called kinetophores?
Yeah, and you could this one right here. I don't know if you can see my mouse. Can you see my mouse moving?
We can.
This one's kind of a good example of how you'll have the stalk, and then it'll produce a ball at first, but then the ball sort of opens up and produces like these huge, dense stalks of spores. If you were to zoom in on this star that kind of exploded, each one of these is just thousands of spores attached to a little stalk.
Mm-hmm.
So millions of spores produced on this plate.
It's pretty. One of the microscopic time lapses I was able to take through the stereoscope was of koji mold. It didn't look like this, but it definitely looked like almost like little dandelion flowers once they opened up. It was beautiful.
Yeah. Yeah, so here's what they look like when they're younger, right? They have the stalks, and then the kinetophores are closed.
See, to me, man, that almost looks like it's hanging upside down. Like that looks like it's hanging, not that it's standing. It's crazy that it can stand like that.
But as they get more mature, they open up and they produce these big spore-bearing structures.
Oz TV just says aquatic fungi. I do know that you have some aquatic fungi somewhere, and we could definitely talk about that. But I do want to go through these.
What's the indigo blue one?
Indigo blue one. Oh, Samantha's dying to know the blue one. Oh my gosh! Look at that.
That's gorgeous. It's so
There's
pretty.
a yeast-like fungus. That's kind of wet in appearance. Also very slow growing. All right.
See, that one was found moss on a granite boulder.
Yeah, some of these I think were samples I got when I went out hiking on my own.
Yeah, sure. What about that one right there? I think that's the one Samantha's dying to know about.
This was cool, right? Because remember I said earlier, if you see blue and purple, then that's kind of interesting.
Oh yeah.
So this colony, when it got really mature, started to produce this like bluish, and then goes to this like deep purple brown. I was like, I've never seen that before.
That is crazy.
The other was kind of boring, and this photo does, like, the lighting doesn't really show it. Like, you can't really see the blue here. But also, I noticed that here you can kind of see a little bit of the blue around the edges.
Yeah. That's neat.
It's fascinating. That one was found on the Mossy Creek, Samantha, as well.
Oh.
I saw that when he was pulling it up.
And this one's interesting. Like, you could look at this if you didn't know any better and be like, okay, what's the big deal? But they're on some species, they'll grow out on the edge, and you can kind of tell like something happens morphologically to the mycelium. Some of it stays aerial, some of it goes subterranean and doesn't want to be above ground as it goes out. You see the patchiness?
Mm-hmm.
That's sort of unique to this particular organism.
Reminds me a little bit of the Hericium plates.
It does, yeah.
And there's a reason for all that, some evolutionary reason why it developed that capability.
Just don't know. Like, I wish I knew what it. Like, Hericium kind of confounds me the way it'll grow through a plate and then just create little tufts of. I forgot what the structure is called. It's even before Primordia. Hyphal knots. There we go. And oh my gosh, that looks like a sandworm's mouth, or something else I don't want to say, but
I have a friend. It's like, that looks like a cat's butthole.
That's exactly what I was thinking. It's wormy.
It is weird though. Like, it's dark in the center and produces these kind of like they're folds, but they do look kind of wormy.
Yeah. Have you ever grown golden oyster before?
I'm sure I have. It's been a while.
So golden oyster mushrooms, what's interesting to me a lot of times is that not every culture does it a lot, but where I started getting wild samples sent in to me from like Indiana, Ohio, New York this year, they'll grow kind of like that, where they'll grow and then they'll turn dark in the center. Like a dark, like I'm like, oh, the plate's dead. And it's not dead, but it's just these black and brown spots in particular that's from the lime yard.
Yeah, that's from the lime court. And this is potentially a new species.
I really did not expect you to get anything out of that lime pool. I thought nothing is going to survive in that.
I mean, honestly, I took so long to process the samples that if I would have processed them like a lot sooner, I probably would have got a lot more stuff. But they like sat in my fridge for 6 months before I had a chance to get them.
I mean, 6 months and you're still getting treasure though. Like this is what I love. You were talking about this, about how, you didn't have access to the mushrooms and stuff so much, but like the molds, like I assume where you were going with that was like, it's everywhere, right? Like everywhere. Yeah.
That's it's everywhere. Mushrooms. And it's in places people aren't looking. Like, how many people are going to a lime quarry and sampling water? And there's very few people in the world that have the time and interest in doing that.
Or catching fleas.
Or catching fleas.
Yeah, catching fleas. So here's the DNA sequence for this one.
So that's the barcode a barcode ITS that you got. Yeah. Yeah. And then, like, if we wanted to get a full sequence of that, not saying that we do, but what would we do? Would we how much would that cost us? Where would we send it off to?
Well, we could sequence a couple of other genes for fairly low cost. The same amount that it costs to sequence this is probably where I would start.
Okay.
But if you want to do like a full genome sequence, it's about $1,000 to sequence the whole thing.
Okay.
So, and what's
How
cool
much are
about
the
this?
Machines?
So it depends, right? If you want to do the actual sequencing yourself at like the physical sequencing, you need the only option that's really tenable is using a nanopore. And that's what Stephen Russell's using. That's what Alan is using all his samples with Stephen Russell, and Stephen's using the nanopore. The startup cost for nanopore is around $5,000, and then you have these replaceable nanopore. I can't remember the names of them, but they're about $1,000 a piece, and that will let you barcode about 1,000 samples or fully sequence like a few different fungi. So the cost is, it's reasonable. It depends on like how invested you want to be into it. I tell people it's like not any different than like buying a MacBook or something like that if you want to get into sequencing, But the cool thing about this one is it's in the order Heliotialis, which has all these really cool cup fungi.
Wow, look at that.
So it's probably a cup fungi, which is why it's creating such a cool morphology.
I mean, heck, the green elf cup I'm growing right now, the only way I have found to grow any amount of biomass with any speed is to liquid culture it. That's the only way I've found.
I love how it matched the tube holder in the background.
No, I didn't notice that. Samantha, you always notice the oddest details to me. I've never seen that. I mean, it makes sense. I'm
fascinated by the colors they produce, honestly.
Oh, she has definitely been on that. The different colors. What was it? The one that we just you were just talking to somebody that here at the shop about it, or no, you were talking to Alan about it last week.
I forgot.
Oh my gosh, it's the one that we just cloned out.
Talking about the spalting strains?
No, the big orange one, Cinnabar bracket.
Oh yeah.
Samantha, so like,
okay.
Yeah, like it's.
I've got a culture of that, which is it's orange even in culture, which is cool.
It's beautiful, man. And so apparently Samantha learned that there's someone somewhere using it to produce vanillin, and then there is apparently a way that you can use it for dye as well. So, which is pretty cool.
I mean, now that we have like ChatGPT, one of the things I almost always do is once I get the species or genus, I just hit up ChatGPT and say like,
plug it in.
Are there like what natural products have been produced by this genus, or how is this used, technologically?
I use ChatGPT almost daily.
Oh yeah, definitely.
It's super helpful for that.
I have limited time, but I'd love to answer any questions if anybody wants to.
Yeah. Oz TV's asking, what media do you use for molds? And then he said, what EPD?
MEA, I put it in there. MEA, malt extract agar. That's kind of common amongst mushroom growers, and then potato dextrose agar as well. I use that because I'm one guy, and if I start trying to like tweak with media for the type of work I do, I can get lost in the world of media. Because that said,
it's easy.
Occasionally I will get invited to go to like an academic lab, and they'll be like, "Oh, we're working with blood agar, or we're working with this exotic media," and I will be like, "Great, can you give me like 24 plates so I can take those back and see if I can isolate any of them?" And so I had a professor give me some ultra low pH plates that are called bile salt media that emulate the salt in your bile.
Oh gosh.
Yeah. And I isolated some cave fungi on that bile media and got some really cool stuff there that are I think they're called halo tolerant. They're extremely tolerant to high salt environments. So you can use media to be much more selective to find odd and unusual things, which is helpful because if you start out doing mold bioprospecting, your biggest problem you're going to run into if you don't have a trained eye is like the fast growing molds are going to overtake all the cool stuff. So a large part of what I do when I'm first doing an isolate is I'm constantly looking at it under a stereoscope, and I'm looking I can almost identify certain molds like almost immediately when they start growing, and I just cut them out immediately so that I leave the slow growing stuff, to grow and then isolate them
Just like-
away when.
Needle biopsy?
I don't do a needle. I just take a scalpel and just cut a small square. Not even that.
He's not trying to subculture it. What he's doing is he's removing the stuff that he's like, oh, I know what that is. So he's trying to cut, like, you want to cut a wide berth around it so that you're getting any tendrils that are growing beyond.
That's exactly right. I just cut enough of a margin where I'm like, I've got enough. I've got enough of this cut out that I know won't grow and take over all the other stuff.
Because, like you're saying, this quick growing fungi, I mean, they've got the initial advantage for sure. So
it's the aspergillus, penicillium. It's all the soil. A lot of soil fungi. They're just so fast growing, and it's like you can leave them there, and if you don't look at them for 2 days, they've overtaken the plate, and you don't have any other, like, there could have been 20 other things that were really interesting, but you never got to see them.
I have definitely had that problem with bioprospecting, even mushrooms, because if like I nick the outside edge, or let's say I'm dealing with like something that's really thin, I oftentimes will have contaminants in it, and if I don't get like a green elf cup, can be kind of hard to clone, and so you get in there, you nick the edge, and you get something fast growing. I can't tell you. I've probably lost 5 or 6 strains this past fall just to the quick growers. Nowadays, I've gotten to the point where if I can get to a specimen and it's fresh, I pretty much have that in the bag. That's the nice thing about macro fungi, is I can get to the sterile tissue. But my problem is worms. Worms drag bacteria all through a mushroom. So it can be quite... Mites are another one. Mites, beetles, but the beetle larvae are probably the most destructive.
We notice a lot of clover mites on the wild strains.
Say what?
So we notice a lot of clover mites and stuff on the wild strains.
Speak to the microphone.
Clover mites? Noticed a lot of clover mites on the wild strains.
I couldn't hear. Dang. Funny. Definitely, like, mites are a big problem. That's one of the reasons why I take the time to seal every single dish I've got, because I know people in a lot of labs will like put them on the sticky mats and stuff, but man, mites can destroy an op quick.
I actually don't wrap most of my plates when I'm working with them. I just let them sit in front of the lid with the lid on. As long as you don't open the lid without the hood on, you shouldn't have any issues with contamination, unless you got some serious like drafts that are coming through that are like, but
Yeah, I do almost all of my mold work. I've got a side table away from the fan where there's no breeze. Yeah, that's about the only place I can do it, because I was just sitting there going, if I open this dish with mold, it's going to go everywhere. So, in fact, I've saved a few strains where even if a lot of times people will tell you that if a mold colony sporulates in a dish with your mycelium, that you can't save it. But I've been able to chase it out pretty quick, especially if I go to like you were discussing in the plates, like going with different agar nutrition types for selective media. You can do the same thing with the woody fungi, just by doing some sterilized sawdust in a dish or something like that, and then growing on that. And the mold has a lot harder time breaking down that sawdust usually. Not always, but usually.
A lot of people will recommend water agar to as a sort of selective medium for doing isolates, because it does provide a carbon source. The agar has carbon in it, so the fungus can grow, but it's not super nutrient rich. You would think like, wow, I was surprised that anything could grow with water agar. But it can, and it can buy you a lot of time.
It can, and
we clean up cultures a lot of the times.
Yeah, we'll clean them up with water agar sometimes. I know someone who, he literally puts liquid culture every time he gets it from myself or anyone else. He literally puts it straight to water agar just from liquid culture, and I really didn't think there'd be enough. I was sitting there thinking, if you put an agar wedge on the water agar plate, okay, it's drawing from the agar wedge like a fat reserve, but like liquid culture, I was like, I can't imagine how it does that, but it's the carbon. I will say, I've also noticed a big difference. People always say to use distilled water or whatever when making your water agar, and I was like, yeah, whatever. So I used tap water, and apparently tap water has stuff in it that helps things grow. So because it definitely doesn't work as well as the the distilled water.
It works better or it doesn't work as good?
It doesn't work. It helps other things grow, but it doesn't help you with your wanting to clean up as much. I mean, it still helps.
It's got all the the metals and minerals from the pipes that it traveled in, plus whatever stuff was poured into it at the treatment facility.
Yeah. Plus, we have, I mean, gosh, lime. We have so much lime in our water here. Huge deposits. Like all of our heaters and our water heaters and our pipes, and I mean, gosh, it's just rough. We get
Happy murder water.
Yeah, which I don't think if they're taking it through the water treatment plant. I don't understand where all the lime's coming from, but it's when I called the city, they were saying like that the our water reservoir for the town is literally at the base of our mine, which sounds suspect to me.
That's pretty cool.
Oz says spores germinate in distilled water, mushroom spores. So I believe that, but I will say that I made up a spore syringe the other day, just using sterilized water, but it was sterilized tap water, and I have a fairly thick mass of mycelium growing. Like I just put the syringe aside and didn't touch it. And then picked it up the other day and I was like, oh my gosh, there's like a thick carpet of mycelium in this water syringe. It's just spores and water. So it, and I don't know, I'm wanting to put it on a plate to see if it's different colonies that are just kind of holding on to each other, or if it's literally like all the spores have just decided that, okay, this one parent wins or whatever. But I don't know much about that kind of stuff. Not yet.
Yeah, some experimentation to do.
Yeah, oh yeah. I'm gonna plate it later. The problem is separating out the mass from the water without jostling it too much where it breaks apart. Because if I can just get it out in one little thick strip, usually what I'll do if I'm trying to do something like that is I'll push it out. Like if I'm doing liquid culture to a plate to test it, I'll do a few drops in there and then find where the mycelium is, and then try to suck up the liquid culture with the needle as much as I can, so there's not a bunch of liquid in the plate. But real quick, I know you're about to go. How do you store you said you have like 500 species, strains? I don't remember what you said.
Yeah, strains. Most of it's deduplicated, so most of it is new, different species. So
Okay.
How I store them is distilled water.
Distilled water? So you're doing the distilled water in the vials?
Yes, in PCR tubes. I mean, I actually have triplicates of everything. I have the original isolate on agar that I seal it, I put it in a baggie, and I put it in a box.
Okay.
Depriving it of oxygen after it's grown will kind of just stop all growth basically, and it'll just kind of be frozen there. And if there's any spore, like if for some reason the water dilution doesn't come back to life, I can always go back to the plate and almost always revive it that way.
Okay.
There you go.
But basically sterilize some water, pipette it into PCR tubes in front of a hood, and then take a sterile toothpick or something sterile and just swab a piece of tissue or spores into the tube. And you want to fill the tube up enough to where you minimize the amount of oxygen at the top of the tube. So you don't want to have like half water. You want it to be almost all the way full. But then I put it into a box for PCR tubes with that. They're labeled. All of the tubes are labeled with their strain number in my database, and just store them at room temperature. And when you go to revive them years later, you can just pipette, resuspend it, mix it, and then plate some of the tissue.
Okay. And so you don't even do like agar wedge in distilled water. You're doing just like a tissue scraping.
Sterile tissue scraping of the tissue and spores into a 1.5 milliliter tube, as OzTV just suggested, a PCR tube.
Yep. Okay. And then what, you just sterilize those in lots, or?
They come sterile, but I do sterilize them. I'll put them in a mason jar and sterilize them.
Okay.
And then I'll just open the mason jar, pull out a tube, pipette sterile water in it, and then put some of the tissue in there, label the tube, and put it away, and it's good to go. It's also an easy way to share samples with people too.
Yeah, I was going to say that you're the one that actually. That's why I asked, so you could tell people, because you shared some samples with me in those little tubes, and I was like, "What?" I still have not gone to that method yet, but I keep thinking I need to, because Dr. Ed Grand, that was in here earlier, talks about that method. You've talked about that method, and I've just always been scared to leave the slant model. But it would be nice, because it would take up so much less room doing it that way. And from what I've heard from everyone is that it actually seems like it's a better way to go. Like the stuff seems to last longer. So,
well, slants are good too. I mean, slants they don't take up a lot of room. They don't take up much more room than tubes, but they're more expensive. If you really are serious about a collection, you probably want to have both, right?
Yeah.
And if you want to share it with somebody, the water one is really easy to share, right? Because you can just pipette. You don't even have to give them the full amount. Just suspend it and give them a little bit of the suspension. Send that to them, and then they can just plate that. No,
when you say suspend that, what do you mean by that?
Mix it up really well, because what will happen is if you put the spores and the tissue in the water, it'll settle to the bottom.
Mm-hmm.
So before you go to pipette it out to share it with somebody or plate it, mix it really well, or use the pipette. It's called resuspending, where you kind of pump the pipette in and out so that everything gets homogenized into the liquid.
Yeah, we do that with our liquid culture bioreactors all the time. We'll sit there when we're pulling syringes or whatever. We'll pull one and push and plunge a couple times before we pull.
Definitely don't freeze it though. You can just store it at room temperature. You do not want to freeze it. Freeze it will kill it.
How long will it last at room temperature?
The most I have samples from 6 or 7 years ago that I've recently revived.
Okay.
But I got it from I found some professor did this for his collection. I wish I could find the website, but he did a whole page with photos and a write-up on it, and he's like, it lasted 15 plus years for him. The length of the time he was at his research, he's like, maybe there were 2 or 3 times out of the thousands of samples that he had where it didn't revive. But I think if you've got a couple out of a 10,000 collection in sterile water that don't revive, that's a really good success rate.
Those couple are statistically insignificant. So
it's like the one contaminated bag out of 300.
I was saying vortex. I assume he means like a vortexer. I've got one that I use in my lab sometimes.
I don't have a vortex. I just use my pipette to just pump it and resuspend it. But vortex will work too.
Yeah, that little vortex. It's cool. It's just a tiny little thing, and you just it turns on when you push on it. So if you push one of those little tubes in, it'll just. It's
those are cool. Yeah, I mean you could make an argument that sometimes might be aggressive and could cause some cell death. So you don't need to do it that aggressively. You might just want to. You could just kind of do this with the tube.
Yeah, that's what I was gonna say. That vortexer is super cool, but I've used it maybe once in the past year. The rest of the time I just shake, like you're talking about. Just gentle shake is usually enough. The one time I had liquid culture syringes that were pooled and then set for almost 3 years, and I used the vortexer, and even that wasn't enough to homogenize it. It basically just took the liquid culture and twisted it into a rope, and it was just okay, this is stupid. What worked out really well though was plunging the culture back and forth, and that broke it up. But as Fungi Florida said, Castellini's method, is that?
Yeah, that's the one.
Okay, that's the water storage.
Yeah, and you can find tons of again, ChatGPT. Say, give me a protocol for Castellini's method.
There we go.
It's very low cost. It'll spit out a protocol for that.
Yeah, that makes sense to me. Well, it seems like we've caught up on the questions and stuff, Josh. I know you're probably wanting to head, so I appreciate you hanging out with us.
This was a lot of fun. Thank you guys for inviting me. Like I said before, it's kind of surreal, and it's really cool to come full circle and be able to hang out with you guys.
Absolutely, man. I mean, it was
Likewise.
fantastic for me. I always like talking to you. I'm hoping that I can get up to you at some point.
Definitely. My buddy Connor, who was a former mushroom grower, I don't know if he said he was going to listen. He's definitely excited to meet you, and I think we might be planning a trip maybe this summer or spring to come and do another bioprospecting trip.
That sounds good to me, man. I've got this spring I will be wanting to do. I'm trying to catch up on everything because I've got a bunch of stuff plated from the last bioprospecting season, and where people have sent me stuff, I've quickly become overwhelmed. So I'm still cleaning up and slanting stuff from this past mushroom season, and I'll probably be getting finished right about the time the next mushroom season starts. When you guys get out here, there's a couple of islands I know that you may want to hit. And I've got a buddy with a boat, or we can just take the canoe out. Whatever, the canoe just takes longer. But there's Bird Island here. Literally, the birds have migrated here. Like National Geographic came out and did a story about it. That's hopefully supposed to air soon. But like thousands of birds come to this lake, this island, and it just started the past few years.
There's poop everywhere,
and it is like a carpet of bird crap. So we can get you egret and
love the poop. Love the poop. Offer still stands on the guano droppings from your son.
So. We're going to get you some guano droppings, man. It's funny because Samantha got mad at me. I was testing rabbit manure for growing like manure-loving species, like portobellos and things. And so I took it and I was like, oh, well, I'm going to do a moisture content test, right? So I microwaved it and oh my gosh, Samantha immediately kicked me outside and then threw away the microwave and was like, it stunk so bad. I've never had anything like that happen before, so it was. Well, maybe we won't do that in the lab. What I now have a microwave designated for manure moisture content testing, so you just let me know.
I'll just tell you about the time I thought it was a good idea to microwave some E. coli growing in liquid culture to evaporate out the liquid. That was not a good idea. My wife has not forgiven me for that.
Yeah,
I will bond then.
The things we've done to them, I guess. My goodness. Well, real quick before you go, let's plug your socials and make sure people know where to find you. I know that I've got your website pulled up here so that people can see it.
That's the picture.
Basically, EverymanBio everywhere, basically.
Everywhere. There we go. EverymanBio. He also has on Instagram and there we go. Instagram it's just at EverymanBio. So just search EverymanBio. He's hopefully everywhere. If you guys want to talk to Josh about sending in some manure samples because he likes the poo and
not your own.
No, please do not just send him stuff. Talk to him first.
Don't send your own poo.
That's right. Yeah, please don't do that. That's a federal offense, I'm pretty sure. Guys, check out EverymanBio. He's doing some really cool stuff. Make sure you go like what he's doing, and if you guys want to help with the citizen science, there's a whole lot more that we didn't get to talk to about. Basically, just trying to bring science down to the grassroots level that he's doing a lot of work for. So if you guys would like to support him, please go check him out. Josh, thank you so much for hanging out with us.
Thanks for having me.
You should make a moldy space.
Yes.
Instead of a maker space.
Keep sending me mold. We'll have to do this again sometime.
Absolutely, Josh. Can't wait to have you back.
All right, guys. See ya.
Take it easy. All right, guys. Well, that was fun. I really like that. Josh and I, he came down and like I said, we got to go run around. We got to trespass together a little. We got some
jelly.
Yeah, Samantha really was. She wanted to go, but she had to hold the fort down. Speaking of, Samantha, I'm going to let you hold the fort down again. I'll be right back.
Oh, no. Appalachian enoki.
Check that out.
Awesome.
Oh, so then people were saying they couldn't hear you.
What?
Why couldn't they hear you?
No.
Oh no, she's sitting there talking and people couldn't hear. We're missing your
Sad
jolly
day.
good voice, but they see your smile. I have no idea what you just said to everybody, but they didn't hear any of it. I'm sorry. I
okay.
think I know what happened.
What'd you do?
I think I needed to hit a second button when I walked away. Anyways, check out that Appalachian enoki. This thing is crazy.
And there I got excited.
You did. You can repeat it. This thing is fruiting like crazy. It actually had a little bit of old fruiting in there too, a little bit. So.
Yeah. But this was just to kind of see what would happen.
It literally looks like spaghetti.
Gluten-free spaghetti.
Definitely gluten-free. Super cool. What's up, Michael Allen? Sorry, guys. But you couldn't. Oh.
What?
There you go. Now you've got the chat. You could have seen that people were saying.
I'm learning.
You are learning. Cheaper than a college education. What's up, Alan? Glad to have you and everybody else. And yeah, sorry guys for the muted and the sound. I'm glad I didn't take too long. Samantha was super happy. Michael Allen says, "Thank you for the handwritten notes, Samantha." A lot of you guys
You're welcome.
don't see. If you don't order, you don't get the handwritten notes. So
Well, not every order gets a handwritten note.
that's true.
Sometimes I don't have time.
It's true. Real quick, so you guys, I'm really glad that we had that conversation with Josh.
That's what I said.
That's basically what you were saying. It's cool to see the work that he's doing. I know as mushroom growers that we're not always excited about mold and bacteria, but I know that a lot of you guys are like me. And what got me into mushrooms was, I don't know, kind of the variety of things that you can do and study and play with, and mold and bacteria is kind of just a well, I mean, it's just another step from that. I mean, I've gotten to koji culture from that. You know, I take plates, I'll grow them out, I'll let them dry, and then I grind them up, and then I've got my koji spores. You know, all that I need. I've got.
So some of your first plate transfers off of the Appalachian enoki, whenever you were cleaning that up, there was like a really sweet, almost fruity smelling yeast on that.
Yep, that's true.
We isolated that. And so it's a really cool, very fascinating thing to be able to just go about your business, do your everyday thing, and then just have these small little interesting, cool things pop up and be able to do something about it.
Absolutely. Yeah, one of the things that I did, was it last summer? No, summer before last. I took blackberries. My son and I went blackberry picking. Just rode the bikes up there and then started picking them. And then I brought some blackberries back and took them and rolled them around on petri dishes. Literally just dropped the blackberry in there and rolled it around, and then dump it out, close it up, do a couple more dishes like that, and then I isolated this yeast that I brew with occasionally. I will tell you, it makes a sour brew. Like, it is sour. It's got alcohol. I found that out New Year's Eve. It definitely makes alcohol. But it's
well, that's the one. That's the one where you wrote "I love you" on the petri dish, and then you made the sour kind of wine out of it, and then it was like sweet and sour.
That's true. I was like
the most romantic thing ever.
I did a yeast swab, and I just wrote "I love you" in the plate, and Samantha thought that's super sweet. Like, she's like, "Oh my gosh, I can't believe you did that for me." I was like, "Yeah, it's sweet, right?" But then the wine turned out to be sour instead of sweet. I mean, it was a mead, so there was the sweetness to it still with the honey. But I too, Oz, I love edible mold, like cheese mold. Blue cheese is my favorite. In fact, I just found out, Samantha and I, every Thursday, so we're about to leave here in just a little bit, and we're going to go down to the Mad Ox. And every Thursday, at least I assume we're going to Mad Ox tonight, but they have a wedge salad that has got blue cheese on it and just big chunks of the bluest, moldiest cheese. Samantha doesn't like that kind of stuff. Samantha, I made a lacto fermented pepper sauce from our peppers last year. Samantha smelled it and she's like, "No, I can't do it." But a couple of my boys and I, this pepper sauce smells like Slim Jims. I don't understand it. It's a sweet pepper sauce with a couple of Scotch bonnets in each one to bring up the heat. It's amazing stuff. But it definitely has kind of like a Slim Jim smell to it. I don't know why. I know that Slim Jim's kind of like a, it's got a fermented meat to it anyways. Like the probably fermented peppers added to that as well. But
And anything that smells that way, that's not actually what it is, it bothers me.
I love pickled everything. I love sauerkraut. I love, give me like Samantha likes sauerkraut when it's like 2 or 3 days into the fermentation. I like it when it's like 14 days. Like,
it should still taste like cabbage.
No, it shouldn't
because
taste
it's cabbage.
anything like cabbage. It should taste the least like cabbage it can possibly taste.
I don't like cabbage texture with non-cabbage taste.
Nerd Nature just said, "Lol, I just realized the sign says spawning, not spanking." What kind of yeast did you use? I'm very interested. It's I like the sour brews. I have the decent wild yeast. Nerd Nature, just so you know, I don't see stuff as easily. I just happen to come over to Instagram, so hopefully you're still watching. But if you come over to our YouTube and our X streams, I can actually see the comments much easier. I'm gonna have to try to fix this with Instagram. I just don't know how to do it. But anyways, the yeast, I don't know the species. I just played around with it and did a small brew with it to see if it would be good. And if it smelled like a dirty diaper, I wasn't going to drink it. But Oz TV says like FY protein they found in Yellowstone Springs, fungal yogurt. Interesting. So they found a fungal yogurt protein in Yellowstone Springs. That's interesting. Wonder if anyone makes an alcoholic beverage from fermented chocolate. Someone does. I don't remember who it was. Someone actually makes Eric Askew. That we had on the show before Christmas, I think.
I think so.
He sent me something about a crab whiskey. They're literally making whiskey out of these like invasive crabs, and I want some so bad. If anytime people want to send a gift to the show, I'll do an unboxing live. I will happily do it, or I will try your product or whatever that you send. I'll try it live. If anyone gets me that crab whiskey, gold star for you. But you just send it to the address that's listed on our Google page. That's the shop address. Feel free to send us your packages. I know a few people asked us about it, about sending gifts and stuff. And in fact, someone sent me this not too long ago, and I am just absolutely in love with this. So
on a wine stopper.
Let's see. This was I can never remember his last name. He sent it for Mossy Daddy, and it's Michael. I can never read the last name. Can you read that last name?
It's beautiful work, though.
It is beautiful work.
Handmade by. Oh, come on.
Right. I can't read it. But yeah, that's it. There you go. Nerd Nature. Nice. Yeah, glad to have you, man. We're here every Thursday at 4. So please feel free to continue to join us. Excuse me. Do you have LC multi-packs or discounts for multiple orders? I will say, Noah, that I have a 20% discount. I love mold 20. ILUVMOLD 20. Save 20% site-wide. If you guys want to do that on mentorships, I know the mentorship page isn't up yet, but call me and let me know that you want to use your discount code on a mentorship or a consultation. We'll book it for you. I'll send you the PayPal invoice, and I will apply that discount for you guys. So use code I love mold 20. It's a new code every single week. We do it randomly throughout the show. I don't have bulk packs up right now. I'm rebuilding them, but I'm about to release a whole bunch of new strains. And so we're going to do like we're going to retire a few strains. We're going to bring some new strains in. We'll do a bulk pack on some of these, but I don't do bulk packs right now. But I will be doing them fairly soon. So Bummy out Florida said, easy way to test for a wild yeast that can tolerate alcohol is to transfer your wild yeast strain into a sugar water solution, but add ethanol to have a starting point of 5%. Nice idea. I like that. I like that a lot. Yeah. Have you ever tried Dungeness? I don't think I have, Rager. I've had crab, but I don't know if I've had Dungeness crab. I'm not sure what that is. Just subscribe and I'll gladly share. Thank you very much, nerd. I appreciate it. Glad to have you on board. I feel like I know you from somewhere. Like, did I follow you today? Maybe. I'm glad to have you guys. Let's see. What else we got today? Oh, something I wanted to ask your all's opinion on. Let's take a short. What? You got something?
That's the Dungeness crab.
I wouldn't know if I've had that or not, though. They all look the same to me. I do. So I was telling you guys, crab meat, lobster, shellfish, all that stuff's like my favorite food ever. That's absolutely my favorite. Hey guys, do you send shipping confirmations for y'all? Yeah, we absolutely do, River. We absolutely do. We send shipping confirmations with tracking numbers in them. We just got pretty much all of our liquid culture premix orders out. We were waiting on sugars for those. Anyone who's ordered BS26 is still waiting probably till Monday. We just extracted a bioreactor of Fat Man and Appalachian and Enoki. No,
we're waiting on Rocky Top and Pink and a few others.
Did we pull Appalachian and Enoki and Fat Man? Okay, so those orders have shipped or are shipping. And what were you saying?
Oh, we're waiting on Rocky Top and the Pink to grow in as well. So if you ordered multiple syringes, there's some people who went through and ordered like 30 of them. So you're just gonna have to. I can't. Sometimes I will split it in half, but I'd rather send it all at once. So that's kind of the thing right now is all of that. And then there was something else that I was. I can't remember.
It was Rocky Top. I meant BS26. I think is already shipped, right?
Yes.
Okay. Yeah, I'd already pulled BS26, so it was Rocky Top we're waiting on. But that should be ready any moment. So we got that. And then Mother of Pearl still needs 2 weeks to grow in. So, yeah, I agree. I'm everywhere. Any chance you could upload the audio of these to Spotify? I might be able to upload the audio, and I don't know, Peter. I'd like to. Let me check into it. Let me see if I can do that, because I do a recording of these. And it should be pretty easy for me to order that up with a what do they call it? Spotify. There we go.
I remember.
Go ahead.
Memberberries.
So the members.
It was in relation to do we send shipping confirmations on the LC orders? Mm-hmm. If you put your email in, it'll automatically send you a shipping conversation. Oh my gosh, a shipping confirmation. I'm tired and hungry, y'all.
We're going to go eat in a minute.
It'll send that to you via email whenever I purchase the label. And then if you didn't include an email, then what I do is I'll go into your order number on the back end of the website and I'll include it as a customer note. So if you need to, you can go into your profile and check that out. Anyway, sorry.
Don't be sorry. He said, sounds good. I ordered some BS26 to head to, I guess that's British Columbia. Excellent. Do we do phytosanitary certificates with Canada, or is that just shipped without needing one?
I've been sending them to everyone.
Okay, fair enough.
Just to be sure.
So if it's international, okay. I thought Canada and Mexico might not require that, but I couldn't remember. I've got a bag of black pearl and golden oyster spawn that I want to fruit, but only have CV substrate. I just need one fruit to get spores for grab and drag monos. Do you think it'll fruit? It'll absolutely fruit on CV. Absolutely. I forgot what the V stands for, but C is just cocoa core, right? Or V is vermiculite. You probably don't even need the vermiculite if you add just enough grain. But yeah, that should be fine. Thanks. I was a commercial winemaker for over a decade and cultured tons, probably literal tons of yeast.
I'd say.
Ed got me into brewing with his red Drizank vid. Sent me down there. What is red Drizank? Is this like Edward Grand Ed? Like Dr. Ed? I'm going to have to check out red Drizank. Okay. Let me write that down. Red Drizank. That sounds like slang. Drizank. Red Drizank. Okay. Well, I've been down the alcohol rabbit hole for 24 years. So I actually considered UT's got a program for brewers that Samantha wanted me to go to back in the day, because one of my favorite things to brew is a beet wine, and I haven't done it in years. I need to go buy some beet.
I do not drink, but I like that one.
Yeah, Samantha's pretty much a teetotaler, but she will drink my beet wine when I make it. And then I'll make what I call Beatrum. And this is actually I got it from my family, apparently. My great grandfather's winemaking journal or whatever, or distiller's journal. He had a thing where he did one out of beet, and there were a bunch of ingredients I didn't recognize, like a Cruft of Luft. I have no idea what a Cruft of Luft is, so I didn't add that. But
it's a Cruft of Love.
It's Cruft of Luft. And I assume that's what he wrote, because his handwriting, it was old and tattered and whatever. But they used to make a distillate out of beets in my family for some reason. They made it with corn too, but I mean, typically, like, beet seemed to be the most common. So I modified the recipe for as best as I could figure. You know, like I said, some pieces were just, I had no understanding what they were. But it turned out to be really good. Super good as a distillate. Super good. Clear or aged. I actually prefer it clear. And I call it Beatrum. But let's see. Samantha says, looked up, Cruft is a term used to describe something that is redundant, defective, or useless. Well, that's not helpful.
And Luft.
And Luft
is a Norwegian word that translates to air or sky. So air.
Useless air.
Useless
a useless of air.
I thought maybe it was like a crust of loaf, or like an old sourdough piece or something like that.
Right. And Nerd, you said sugar beets. I have no idea, but probably not, because my understanding, my family, I don't know if they had access to sugar beets back then. It would have been something like the purple beets, or what do they call them? Bengal beets, the fodder beets that are used for animal feed. Either one, both of them are high sugar. I mean, not as high as sugar beets, of course, but yeah, I don't know. It'd be interesting. Hibiscus wine, yeah, Dr. Ed. Okay, yeah, I figured. Anyways, the beet distillate. I love when someone says you don't have to use verbs. Think of that stuff. The beet distillate, I made the I don't know what you call it in that one, because it's not a beer. So, you know, the pre-distillate, whatever the wash is going to be called. I just was like, oh, that's an electric purple color. It's beautiful. Like, it's
very pretty.
Really beautiful. Once you add the acids to it, it just turns from a dark mud color to a purple, and it's super beautiful. And then you just drink it like a wine, and oh, it's so good. When you make it sweet, it's like sweet and earthy, and it's super easy to make. And yeah, anyways, it's beautiful. But I just use grain and core and masked mofo to keep it simple. Connor says it would be probably faster to name what Mossy Creek oysters won't fruit on. True that. I have fruited. Well, okay.
There's a trick to that.
There is. As I breed really good, really aggressive stuff, but I have fruited oysters on. And please, I'd never served this to anyone, so do not say anything bad. But I totally grew them on Madagascar Hisking cockroaches. I grew them. I've grown them on dog food now. I've grown oysters on a dead mouse one time that I found in the operation. I think when the cats killed it, and the mycelium took right to it, and which is terrifying if you think about that. And what else have I grown it on? I've grown it on some crazy stuff. So yeah, Mossy Creek oysters will definitely fruit on stuff. Wow, when you lean like that, I can hear my voice echoing through because your headphone goes right here, and then I can hear. Oh my gosh, that was weird when I did it to myself.
Echo within an echo.
Blue Drizank equals blue alkaloid water made from old spawn. I got a video on YouTube. Great way to use spawn that's too old to fruit well. Nice microdose drink using the acidic juice, spawn, and shake well. Interesting. Okay. What's the red drink? Kind of a ghetto lemon tech. No need to make fruit, so stays under the radar. I gotcha. Edward Grant, I just found some cool bricks of pressed Dry Masters mix blocks. It's packaged like core. They contain methyl donors, beets. Very healthy. What are methyl donors? I don't know what I know what methyl is, but I don't know what donors are. But I will say that what's interesting about the beet distillate in particular to me is that it has a very strong vanilla note to it, and I don't add any vanilla to it. So it's red drizzling is the hibiscus wine. You said that earlier. My bad. But yeah, like I've not had any. I do want to koji beets and then try brewing those as well. So I did a koji last year where I did a bourbon recipe, but instead of sugar wash or malting the grains, I koji'd them instead, and then did that, and that's fermenting right now. So that'll be interesting to try that with what the Asian sacrificing method is with the southern United States recipe. It'll be weird.
Something that's really interesting and fascinates me about koji is the fact that whenever you kojify something, it makes the nutrient profile all the more rich. Mm-hmm. And I think that is really cool. And it makes sense because it's breaking down things it's like pre-chewing it for you.
Well, it takes proteins down to their amino acids. It takes sugars down to the word escapes me all of a sudden. But anyways, it is cool because koji at certain temperatures and feedstocks will produce amylases or protolases, and so you can. It's a beautiful organism because you can make soy sauce with it, and you can make wine with it. So it's neat like that.
Twofer. One
of them twofers. I cloned a chicken of the woods in September. Should it be light orange, yellow on agar? Absolutely. It seems to be smelling fine. Have you ever inoculated a log with chicken of the woods? I have. They will grow. Look up pony log culture. That's a good one to go with. Also, if you want to donate that strain to the Mycologium, I'm always happy to trade strain for the Mycologium. In fact, how about we discuss that? Sorry, Samantha, I just blocked you out. The Mycologium is our program where we are our bioprospecting program, where we partner with people. We partner with people, and you're welcome to send strains in that you have bioprospected. So if you've cleaned it up culture for culture, I'm happy to trade you, swap you culture for culture if you've got a unique strain, especially unique to your bio region. And that's pretty much anything useful. I mean, turkey tail to reishi to oysters, hericiums. If you've bred something that's just really unique, that kind of thing. I'm always happy to do it, and then I'm happy to trade you strain for strain, as I've said. If you find a mushroom that you want to donate, you can always 2-day mail it to us, try to cool pack it or whatever, throw it in a Ziploc with a little bit of cardboard or paper, and ship it to us. If I can get it clean, then I will happily send you a plate back. But this program is where we are trying to save as much of the biodiversity as we can as urbanization and just change is happening. So we want to just collect things, put them back, and then over time make these things as available as we possibly can. So like this past summer, I got several golden oyster strains. We got several chicken of the woods, hen of the woods. I even got a shiitake sent to me from Gold Beach, Oregon, which was a naturalized shiitake. They had never grown shiitakes there before, but there was shiitakes growing. And it's definitely a shiitake mycelium. It smells like it. And browns and everything else. I'm about to try fruiting it. But we're collecting all these strains, so we're happy to share the biodiversity that we are collecting. And if you're please send your samples in. We would really appreciate it. And it'd be nice to be able to save some of this stuff and put it back in a culture library somewhere.
Well, and something else we've been doing too is if there's a mold that's on it, Andrew is cleaning up the fungus part, of course. But then anything that's kind of extra, we've sent samples off to Josh McGinnis from that. So
yeah, if there's
that's
weird
been,
stuff or whatever,
yeah,
like
so
I've
it's
got.
kind of like a 2-for-1 sort of thing. Like,
Also, if you send me like an email with the photos that have the GPS coordinates in it, I'm oftentimes able to send at least a small sample dried to like the Ohio biodiversity, fungal biodiversity project, or one of the places where they're categorizing where these things came from, and trying to just catalog all the species that are growing everywhere. So, we would really love for you guys to join us in that program. Apparently I missed a couple things. How long does your golden enoki take for fruiting after colonization? It's going to depend on temperatures and everything else, but I would say that golden enoki is somewhere around a 6-week turnaround from the time you plant it. White enoki tends to be about a 2-month turnaround, so 8 weeks, whereas the golden enoki seems to be a couple weeks faster. CVS has cool little Q-tips travel containers for $1.69. Nice. Interesting. I always just buy sterile swabs off Amazon that come packed in individual packets, but Q-tips I think are normally pretty sterile. I saw those, but they can't buy them here. Just need one fruit for spores. Going to get the gourmet world on the Mon Daimon train. There are people working on it. I know Main Cap and Stem is working on it. They're going really whole hog, I think, into trying to build a mono library. So, but they're trying to do it more from decaryotization. So, wow, and I thought I was unorthodox. Yeah, I mean, it's a spectrum.
I try to keep them straight.
It's hard. I'm coming out crooked. Could you get lion's mane or oysters to grow and use coffee grounds? Absolutely, Michael. They do not typically like it. The only gourmet I have found that really takes the coffee grounds pretty quickly is shiitake. Seems to like the high acid environment better than the oysters do. But
you did try King Blue.
I've got a King Blue strain that I've been putting on coffee for about the past year that rips through it pretty well. So I've got 2 or 3 strains that I'm about to start training. It does take time to get it trained to the coffee grounds.
It took you like a year, right?
Mm-hmm.
To get it trained over.
Yes, that's what I was saying.
Everything takes time.
It does, and I'll probably will release the Coffee King Blue fairly soon. So, epigenetic stuff to turn. Okay, that's the methylation of DNA. Enzymes. That probably was the word I was looking for. I can't remember now even what I was talking about. But at Michael Meacham, oysters, yes, but lion's mane didn't seem to like it. I got PE and GT to grow in it, so it takes a super long time. Yeah, the coffee. It's just certain things are not going to like it. Certain strains will like it, and like I said, you can train things to like. Like I've got, a few of the manure lovers that I've been able to get to really love wood. Where normally they don't take to it at all. I've got some of the oysters that I've got to be able to take to acid environments like coffee. And this is just opening my eyes to, okay, we can start training things to cigarette butts. I saw someone do that one time. You can train it to a lot of different things. There's a lot of potential in fungus. So I love the HP Lovecrafting logo. Thank you. I had the AI help me with that. So, and then Samantha's going to clean it up later whenever she, one of the mini projects that I've thrown on top of her. So, looks like my kind of cult. The Mycologium is absolutely your kind of cult. I found a super cool phenotype in one of my blue oyster bags. Do you have any recommendations for someone who hasn't isolated anything before? So you've never cloned before at all? The biggest thing really is to just tear. When you tear your let me put the comp their way so you can see a little bit better. Take the mushroom and tear it in half. Don't cut it, and tear it towards your hood. If you're using a still air box, then it doesn't matter as much. Just with a still air box, move really slow. Try not to create big gusts of wind with rapid movements, that kind of stuff. Do not put alcohol in there and then light a flame in there. You will blow up your still air box. I never did that and have never had plastic embedded in my face because of it. I quickly switched to bleach after that. But the
You live and you learn.
you live and you learn. Cheaper than a college education, even with the emergency room visit. So the I actually didn't go to the emergency room. It wasn't that bad. The whole idea though is to peel it open towards your hood and just really try to cut. What I like to do is cut at an angle. So let's say here's the flat. You've got the mushroom, but here's the flat from where you tore it. I like to cut at an angle. So and I like to dig like a little canoe out of like the stem or the cap. And basically what that does is it allows you to free it from the mycelial bonds underneath it. And that makes it a lot easier to select out. It was using a sharp scalpel. I can get away with a charred up, jagged scalpel sometimes when I'm just cutting plates. But when you're cutting mycelium in a mushroom, it's always going to behoove you just to switch out to a clean scalpel, brand new, because otherwise your blade will catch and just tear. Another thing is if you're having a hard time cutting things, sterile tweezers work really well. You can just pinch and grab and pull, and a lot of times you can just pull little chunks of tissue out, and that works really, really well. I found that works really well with things that are really hard to clone, or hericium, which can sometimes, if it's got a lot of water in it, it might be hard to cut. You're sitting there holding it, you're starting to cut, you're making it squeeze water out. A lot of the times it's easier just to grab a little piece and just pull that strip out of with the sterile tweezers. So that is how I would isolate things. But anytime you find anything interesting in your grow, always clone it and then grow it back out and see. There have been 2 or 3 times that Samantha's like, "Are you sure you don't want to clone this?" And I'm like, "I'm sure." And then I'm like, "I should have cloned that. I should have cloned it. I've never seen it again." So always clone.
Pom-pom hericium.
The pom-pom hericium. This hericium that grew with this weird, there's just one bag out of all the others. And they had these, like it was a comb tooth, and every branch ended in this big swollen, it looked like one of those fruit trees from Rick and Morty, with like a big fruit hanging on the end of it.
Doesn't look Dr. Seuss like.
On a very selective basis, I'm willing to take and sequence Pleurotus species, especially in the Celluloderma sect. Maybe I read that wrong then. Plutus instead of Pleurotus. I'm currently working on some new species publications that should be released soon. That's cool. Peter McCoy did. Okay, that was McCoy. I couldn't remember who had done that. Thank you. Question: ever used pH water like 9.2? I have not, but I would not be surprised if that lime water pool wasn't that high. I wonder. We should have asked Josh what the pH was, but I've not used it. Epigenetic transformation may be important to choosing better substrate. Yeah, I mean, there's a lot to work with that for sure. There's a lot. Cthulhu mushrooms. I'd be happy to send the chicken in. I can include pictures of where I found it and even exact location. I'll start a new plate and send it. Please do. You can just look up my shop on Google. That's our address. It's 208 East Old Andrew Johnson Highway, Suite A, Jefferson City, Tennessee, 37760. You can always come back when this goes to a video and rewind it there. We're 2 hours and 49 minutes in. That way you can rewind it to where you want. But yeah, please do that. And then just let me know what you want. Check out my website. Let me know what you want. If nothing there interests you, feel free to let me know in a letter or something like that when you send the sample in what kinds of things you are interested in. Please give us a return address.
That'll help us remember.
Yep.
Yeah.
And
if you just tell us, we'll have 50 different conversations after that, and we'll forget.
I forget things all the time.
You have some chicken in the woods in the walk-in.
I've got
tree
bags.
itself, but yeah.
Those are the Mother's Day. Those are the ones that we found on the ride with my mom.
Oh, I remember that.
Yep. This past Mother's Day on the bikes. Well, guys, it is 2 hours and 49 minutes into this, and we normally go 2.5 hours. So
So we've been going 3.
Well, we have been going 3, and yeah. So what was it? I think it was Josh McGinnis called me Andrew Rogan. There's just so much to talk about. Like, it kills me because there's so much interesting things to talk about. There's like it literally touches everything, and then every time you think, well, I can in it there, something else comes up, and you're like, oh, that is cool. So, however, I owe you a dinner date. So I'm gonna go give her a dinner date. I feel like a wedge salad and some amazing spaghetti and meatballs is calling my name. So this place has the best spaghetti and meatballs and beer. Oh yeah, last time. Oh, I probably should bring him a slant of the yeast too, because he wanted that. He wanted a culture of the yeast. Anyways,
Lovely.
maybe I'll make him a barm or something instead. But yeah, this has been fun, guys. Fun guys. Thanks, all amazing talk. Have a good dinner. At work, this was nice to listen to. The customers must think I'm a scientist studying. They can hear you listening to me? Holy crap. Don't do that to them. Great show, kiddos. Great stuff. I'm looking forward to next Thursday. I'm looking forward to having you with us, Nerd Nature. Good vibes to you too, Spore Muse. Oh, hold on. Question from the beginning of the show. One of my restaurants wants smaller BO clusters. I forgot that you wanted to. Should I use less nutritious substrate or cut massive holes for more pinning? Not sure. Usually when it comes to the size of your clusters, it is your substrate depth versus how many holes you have. So you're going to have an expected load or expected harvest weight. So, 12 pounds of substrate might give you 2.5 pounds. But let's just say you were getting a pound of mushrooms or whatever your expected flush would be. If you do one hole, you're going to get one pound cluster typically. If you do 4 holes, you're going to get 4 quarter-pound clusters, roughly. It's not exact. You can always control that by going to smaller bags or cups or bottles or whatever you're growing in. But you can always go smaller so that, and every strain is going to have a different biological efficiency. So what you got to do is test it. You're going to have to test it and then figure out, this one grows this much, and then you can range it up and down from there. So
I will say that because we did have a restaurant that asked for smaller clusters so that they could deep fry them almost like a blooming onion.
Mm-hmm.
And this was back when we first started the U-cut and we hadn't quite mastered it yet. And now we do the U-cut and then we peel the plastic up and off and then just slightly let it kind of suspend itself in the air. But if you don't lift it up, then you're definitely getting a cluster set along that cut line. And if you go really deep, then your your pin sets are longer and they're smaller. I don't know. It's almost like because you've opened that cut up, you're drying it out a little bit. And then after you get that pin set, then open up the plastic, you'll get a secondary pin set and you can harvest small little clusters that way. That's what I would do. And then upcharge because you're not getting the finished fruit.
You're having to pick more.
Yeah, you're gonna have to pick more. You're gonna have to pay more attention, and you have a higher chance of losing the bag because of the time frame that it takes between the first and second flush and that potential contamination. Something else too is picking from bottom to top, and then additionally changing your gloves each time you have anything at all that's suspect. Change your gloves often as you're picking. That helps keep your blocks clean too, especially if you're making multiple picks from the same block that way.
Yep.
Anyway, hopefully that helps.
I would agree with all that. Have a nice dinner. Thank you. I'm going to send you some pictures of email. Absolutely, Joe. Please do it. Hey, I talked to you on the phone earlier, didn't I? Hope that shoulder is doing well. Great show. Thank you. Thank you, Connor, for joining us. This live stream is quickly becoming the highlight of the week. Love what you guys are doing. Thank you so much, Michael.
Thank you.
So do you wait to see the fruit before making the holes? My last question I'm going to answer. No, I just cut and go. The mushrooms serve me, not me the mushrooms. I know that's kind of that's a naughty statement in the mushroom world a lot of times, but no, I grow mushrooms because they make my life better, not because I'm a slave to the mushrooms. Jake, you just now joined us?
Please answer your Patreon message.
Yes. Also, I need to get some video of your operation so we can do that on here live. So, crap, I'm also getting a lot of interviews set up too. I'm glad you made it too. We're actually just signing off. So, Love y'all. Thank you all so much for joining us. And
Keep spawning
keep spawning culture, y'all.
Bye.